working design is like a certain comstech as in health initiative. And I have written a 5-year action plan on health for this. Before this, I was working as a chairman clinical study committee in the health department.
Advance.
It's okay. Yes, it's okay. Teacher. and IT specialist. The main component that the clinical trial must have the standard for the design of the clinical trial. This is very, very important. How you design the conduct the clinical trial. When you design the clinical trial, the second important is how to conduct. The third one is when you are conducting the clinical trial, you are monitoring or not. If your design is good, if your conduct of clinical trial is very good, but you are not monitoring what is the safety, what is the efficacy data, then it is a useless. So monitoring is very, very important. Also the data, after monitoring, data have the auditing importance. Then you must record all the information, all the data, either it should be on the paper, on the IT, on different sources, you must record and you analyze the data of clinical trials. And this analysis should be on statistical basis. And the last one is reporting of clinical trials. The principal investigator is bound to report to the IRB, to regulators, and to the sponsors. You must report other components of clinical trials are the IRB, that is the decision review board, or institutional ethics committee, and also the regulators. They are approved the protocols of the clinical trials, either it is institutional review board, or the national bioethics committee, or the regulator of the national regulators. Another one is principal investigator or the investigators and the sponsors. The main, very important is the protocol of the clinical trials and also the IP, that is investigational brochure. And these are the essential documents. And moreover, the essential documents of the clinical trial must be documented, all the data should be recorded. These are the main components of the trial quality clinical trials. Clinical trial is, the first one is very, very important. The data integrity. Very, very important to provide assurance that the data and reported results are credible and accurate. If data is fake, and if the data is not reliable, but your clinical trial design, conduct, and other monitoring and auditing is very good, so it is always useless. Another thing is the protection of the subject. Protection of the subject is the main priority in clinical trials. Just to I must say before rights, I say must respect, then protect the rights and also the integrity and confidentiality of the clinical participants are protected. So I add one more thing: we first respect the participant, then protect the rights of the participant, and also the integrity of the participant, and finally the confidentiality. So I add one thing more, that is respect as a professional area in clinical research, they must know they develop a consensus among countries regarding the requirements to conduct clinical trials, and also to harmonize with each other and rely on the data of each country. So ICET guidelines are very important, and whole global world, they follow the ICH guidelines for conducting the clinical trials. And what is the what is the importance of ICH guidelines? Mutual acceptance of data of many countries. ICH guidelines show the credibility of principal investigators and the validity of the data. The second one is new drug application, that is NDA data, to be presented in a standard format. Otherwise, each country, each institution, each organization, they have their own format. So it's very difficult for new drug information data. So this should be a standardized format, and review time for drug application is shorter. That's why we have the whole data. That's to review the data with standardized format. We can reduce that time. Also, reduction in drug development time. Also, it will it will reduce the cost and use of resources by ICH guidelines. Code of Federal, this is the US FDA. Title 21 FDA, Part 1 electronic records, electronic signatures. You can find these guidelines apart from ICH guidelines. You can find in US FDA 21. And moreover, every country have their own guidelines, and also see and follow that one. Moreover, part 50, the protection of human subject. This is very, very important. The protection of human subject, you must see in detail. Financial disclosure by clinical investigators, and part 56 is institutional review board, which is the board constituted in the parent institution and composed of different having different expertise, and from outside the institutions, and also from the citizens of the populations. Also, the investigation review drugs application you will find in part 312. Where you can find guidelines for clinical investigators, I must say you can find guidelines in US FDA organization, and also the ICH guidelines, also the Pakistan. Moreover, you can find the guidelines of your own country, that is Uganda, Ministry of National Health Services. So you can find guidelines and clinical trial.gov. So there are the different sources, different websites where you can find different guidelines. Just to yourself. Clinical trials at the research site. Important to know the good clinical practices. Inshallah, after lunch, I will give the detail on good clinical practice. So you can be ready to discuss the basic requirement and any expectation about that requirement. Document good clinical practice training at your own site. You are the master trainers here. This is your responsibility when you go back to your own institution or your own ministry. You can train the people on good clinical practice and documents. Inshallah, after lunch, we can discuss good clinical practice. Recognize that sponsors require that you follow their integration of good clinical practice. Key components and principles of the ICH guidelines of clinical trials. The rights and safety of the well-being of the subjects. You must consider that Rights, safety, and well-being of the trial subjects are the most important consideration and should prevail over interest of science and society. This is very important. Just for the sake of business, just for the sake of science, and just for the society, you should not conduct clinical trials because the rights of the subjects are very, very important. You just not make them guinea pigs and animals. You must protect their rights. Clinical trials should be scientifically sound and described in a clear detail with detailed protocols. Establish each individual involved in conducting clinical trial should be qualified physician, pharmacist, epidemiologist, public health specialist with higher education, and it should be well trained and experienced to perform their respective research work. I will discuss shortly about the protocols. It is given in ICD part 6. These are the main components of the protocol. You should follow the ICH guidelines, also the US FDA regulations. The protocol should contain a detailed description of the objectives, purpose of the trial, primary and secondary endpoints. Dr. Mustafa Qadri will discuss in his presentation. Type of the trial design, it is a single-blinded, double-blinded, placebo-controlled measures to minimize bias, randomization, or blinding. This has been discussed yesterday also, and also today, Dr. Mustafa Qadri will highlight these points. I don't want to go to details, just I'm giving the points. A design regimen of institutional product, dosing regimen of an institutional product is very, very important. Detail is given in the institutional brochure. Expected duration of subject participation. It should be 1 month, 2 months, 3 months, 6 months, or 1 year. Also, subject inclusion and exclusion criteria. This is very important. Just as Corona, the inclusion exclusion criteria was the subject should not be less than 18 years, not should be above 60 years. So this is inclusion and exclusion criteria. Another thing is consent form. In the local language, it should be in the language, it should be in the Pakistani language. Even in the different policies, there are different languages. You must give the consent form to the participant in local language. It should not be in English. If it is in English, then it should be in the national language and also in the local language also. This is very important, and you must explain the whole protocol to the subject so that he can understand and he sign the consent form. Procedure for advanced reduction. How in the protocol you must mention how you collect the ideas. I have another presentation of the safety and vigilance of the investigation drug. So I will give the detail in that. Investigations. In Pakistan, we call it the main investigators, we call it principal investigator, and after that, we have investigator. So it is up to the national law of each country. This is very important. You must inform consent of the trials must be clear to the investigator and to the subject also record. This is the responsibility of the investigator to record and report all the time. Also, this is the responsibility of the minister to give progress report to the IRB, to the IRB and institutional review committee, and also the draft. Also, he is responsible to give the safety report to the National Regulatory Authority. For pre-investigator is responsible for premature termination or suspension of trial. If principal investigator or investigator finds that the serious adverse events are increasing day by day, and statistically it is not viable that the trial should be continued. So this is the responsibility of the principal investigator and investigator for premature termination of the trial. Final report by investigator. Our principal investigator is responsible to submit the final report to the IRB, to the sponsor, and to the regulatory body.
So this is the responsibility of the principal investigator. So, the principal investigator should be highly qualified. He should be the physician. He has the adequacy of the resources, medical care of the trial subject. This is very important. The insurance of the subject should be done in each clinical trial. He should be treated within the hospital. He should be given the transport facility if the adverse effect occur in his home. He should take care of his food. He should take care of the traveling facility by the investigator. Investigator is responsible to communicate with IRB. Also, he is bound, investigator is bound to give the compliance to the protocol which is given by the sponsor. He should be know about the investigation drafts and randomization procedure, unblinding. I think Professor Murtabha Qadri will throw light on this topic. Investigators, this is in part 4. I don't want to go into the details. Study subject, IRB, institutional sponsors, societies, regulators, they all are bound to be the to follow and implement the good clinical practice. This is also the responsibilities given of the given to the investigators is documented in these rules, guidelines, and laws. Investigators should be able to demonstrate potential for recruiting the qualified number of suitable subjects in the agreement recruitment period. This is very important. In some cases, principal investigator he starts the clinical trial, but he cannot recruit the subject. So he should be in a such position that he can be recruit the subject. He should be well trained, and his staff should be investigator should train the staff who is involved with the clinical trial. This is very, very important, and have suitable qualification experience. Also, the responsibility relies totally of clinical trials on the investigator. Investigator is the main responsible to conduct clinical trials. Major areas are that personal. I don't want to go into detail. Investigator involvement. He should develop SOPs that include the role of principal investigators. He should document communication with the principal investigators, copy on study related email, and retain the regulatory binding, retain records of the meetings and telephones. This is whole responsibility of the regulators. The sponsors, if I go back, sponsors. The sponsor should, the sponsor is the main, the sponsor is the individual, it is the institution, it is the company or organization who is responsible for the initiation, management, and financial support. So sponsor have the major role in conducting clinical trials, and the sponsor is responsible that principal investigator is qualified enough and he do the agreement and have written agreement with them. The sponsor should obtain the investigation institution agreement, that all should be the responsibility of the sponsors. Another is he should permit monitoring, auditing, and inspection to retain essential documents of the clinical trial. Sponsor should ensure timely delivery of the institutional product, take steps to ensure that the institutional product is stable over the period of time during the study. Another responsibility of the sponsor is he should be responsible to monitor that the rights and well-being of the human subjects are protected. This is also the responsibility of the sponsor. Either he do himself or through the principal investigator or through the CRO. He can directly protect the right of the subject directly or through the sponsor. This is his up to him. The monitor should be appointed by the sponsor. This is also the responsibility of the sponsor that he should appoint or do the agreement with the monitors. The sponsor should ensure that as the eligibility monitor, the monitor acts as the main line of communication between the sponsor and the investigator. This is a bridge. Monitor is the bridge between sponsor and the principal investigator, and he should monitor the rights of the subject. This is also the responsibility of the monitor to verify that an investigator has adequate qualification and resource. Moreover, ensure that the investigator and staff are adequate informed about the trial. The monitor should not, any person who is not trained on the trial responsibility to determine whether any adverse event has been occurred or not. Sponsors, sponsors, I already told you. The last slide is about IRB. Last slide is about IRB, that is investigation brochure. It should contain all the details of the investigation drugs, frequency of treatment, and the effects of the investigation drugs. Last is that is the trial is responsible to himself or to his CRO. This is all about the basics. This is all about the basics of clinical trials. These clinical trials design, sponsors, investigation process, monitors, sponsors, safety monitoring. These are all the basic components to have a good clinical trials. With this, thank you very much.
Thank you so much, sir. It was indeed a wonderful presentation, Dr. Abdul Rashid. Thank you so much. So, we had a training last week on Research Ethics Committee, and I mean GCP, good clinical practice. Combine the knowledge of what we did last time with what you are doing currently, you see is a continuous learning, and everything is actually linking. Thank you so much for actually linking the clinical trials and the most important part of clinical trial, which is the IRB or Institutional Review Board. Thank you so much. Now, to Professor Mushtaba. He has a wonderful quiz for us, and I'm sure we're all ready to take his donors plus the gift he's planning to give us. Thank you, sir.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. So yesterday we had some concepts of RCTs, and because we started a bit late, so we couldn't complete the second part. We'll do that first, because otherwise you'll say after questions are from the second part. So we will do this in the last 20 minutes, inshallah. So let's have my presentation, please. The one on the RCTs. So let me just take it back. We can continue, and there was some concept that maybe I read through yesterday, so we will start right from there. So if you just take a look at this, we went through the evidence hierarchy, starting with all the basic case reports and case series, and going all the way to RCPs and systematic reviews. We talked about the translational research spectrum going from the animal studies to cell culture and going animal studies and cell culture and bioinformatics studies, and then going on to small animal and large animal studies, and then going on to all the phases that Dr. Rashid very eloquently described, phase 1 to phase 4. And phase 2 and phase 3, where the RCTs are usually the issue, is what we're trying to understand in detail what is the methodology. So we talked about some key concepts yesterday. We talked about the fact that there are several different types of trials. The parallel group is the single arm, parallel group is the commonest. But then you can have crossover designs, you can have sequential studies, you can have factorial designs where you combine 2 or more different factors in both ends and combine different combinations of that. And then, of course, you can have what is known as an N of 1 trial, that only one single person takes a single drug and then sees the effect, and then after washout, compares it to something else. So these are the various types of RCTs one can do. We talked about some key concepts. Bias is that systematic error we introduce voluntarily or involuntarily in the design or conduct of the study or the interpretation of the study. We talked about contamination being simply people in the rank in the groups which are being compared, becoming aware of the other side's protocol and trying to follow that, so which kind of confounds or which kind of affects the outcome assessment. We talked about the key concept of confounding, which is a variable which independently affects the independent and the dependent variable and makes the outcome look either bigger or smaller. And we talked about what is known as effect modification, which are factors which are after randomization, such as age and gender, which you cannot modify, which will also modify the effects in various groups. And we talked about the fact that each time we do randomization, you also have to make sure that all the factors, like the baseline characteristics, are balanced, which will occur through randomization. And then you also look at all the core interventions. Whatever drugs they're getting, or behavioral therapy or nursing therapy they're getting, should be identical in both the groups, except the only difference is the active drug versus the placebo, or the active drug versus active control. And that is the key concept to understand. We talked about validity, that whatever is internally valid, good randomization technique, good blinding, good allocation concealment, good analysis in terms of intention to treat analysis, which we are going to talk about today, inshallah. And that particular concept is all about internal validity or internal integrity of a study. But then when you look at the inclusion and exclusion criteria, if you have very broad inclusion criteria, narrow exclusion criteria, you will be able to generalize your finding to the population at large. Each time we're doing a study, we're simply taking a sample from an accessible population, and whatever you determine in there is being applied or generalized, externally validated to the target population at large. That's the whole purpose of this study. If you could study the entire population at large with that condition, then you will not have any kind of error. You will get an accurate result. But that is impractical. So you take a small sample, whatever is accessible, and therefore you sometimes make a false positive, which is a type 1 error, or a false negative, which is a type 2 error. Then we talked about the fact that the interventions can be either drugs, it can be behavioral therapy, it can be drugs or antibiotics, it can be devices, whatever it may be, compared with the other. And the endpoints are actually the endpoint is the endpoint that you're trying to determine based on the classification. The principal study is which can be either hypothesis, that can be difference in the two groups, but in the order which you do, rejecting the null hypothesis and saying that one group is actually better than the other. And that particular thing, the endpoints they determine the sample size calculation. So you can have a primary endpoint, a single random determined, for two major primary endpoints you can sometimes use to get the primary answer. Endpoints are involves
patients costs and so feasibility issues are there. So you collect data, and if you can show some difference even in the secondary endpoints, even though your study is not powered to answer to the secondary endpoint, the sample size is only calculated for primary endpoints, you might be able to get something which provides basis for doing a further study. And so that is like for the secondary endpoint. Secondary endpoints or primary endpoints can also be done in the form of what is known as either surrogate endpoints or composite endpoints. The surrogate endpoints are if you find that we cannot show a hard clinical endpoint by the end of the study, like fracture or for our head, for instance, you might say, okay, maybe in 2 years I might at least be able to show that the bone mineral density has gone down in 2 years. And if I can show that, hopefully that is a marker of future development of fracture. Those kind of things can be then demonstrated in retrospective studies, retrospective cohort based on those. So the composite outcome, which is important to understand, is that when you know that there may be some important outcomes, for instance, one of the examples we are using in this course that is there uploaded on Google Drive is a study published in the New England Journal of Medicine a couple of years ago. It's a CKD study. Is an SGLT2 inhibitor, which basically has become now dapagliflozin and pagliflozin, canagliflozin. These are things which are the routine part of the care of the diabetic, especially with proteinuria, which affects your cardiovascular stroke outcomes as well as your renal outcomes. In this CKD that we're going to be using as an example throughout the concepts that we will be continuing today, we are going to explain how those composite outcomes were. Each one of them was the component of the composite outcome was an important clinical outcome, like development of end-stage renal disease requiring dialysis or transplant, or fall in glomerular filtration rate by at least 50%, or death due to renal or cardiovascular causes. So these are all important. Each one of them is important clinically. So we can combine them as a composite, calculate the sample size to include all of those factors, and if any one of them becomes positive, then you say that the study has reached its primary endpoint, which was the composite endpoint. But then if you are not convinced and you say, oh, there was some not very important clinical endpoints in there, then you may want to do another study where you take that composite and put it on the composite endpoint and do it with other endpoints. Then the concept of inclusion and exclusion criteria, we talked about it. The fact that if you want to include people with a specific condition or interest of the importance that you want to include, have to be included based on whether you're likely to find an outcome in that particular group, and if those people are accessible to you, it's safe to conduct experiments on that particular group. So the inclusion criteria have to be balanced against the concept of variability. So you try to keep the inclusion criteria relatively broad, but if you make it too broad, there will be too much variability in the sample. So there's a balance in what you can include, and the exclusion criteria you exclude people from a study if they are eligible. Otherwise, just like the inclusion criteria, but you don't have to include them because of safety concerns or because of confounding or because of poor implementation or because of feasibility. So, the criteria, but if you keep the inclusion criteria too broad, then the inclusion criteria obviously will be narrow, right? So, you will be not able to generalize to the population at large the results of the study. That's the concept of external validity for generalizing. We talked about the fact of recruitment. There's a proper way of advertising for candidates, and that advertisement also needs to go to the IRB for Google. You should not try to say that what you're doing will actually benefit the subject, or what you're doing. You should not offer a new financial incentive, and the advertisement should not try to claim that this is standard therapy. It has to say that you are going to invite it to be included in a clinical trial, and that you will be compensated for inconvenience if the compensation is too big. It's not fair to the subject because it compromises that autonomy and raises an element of perseverance. So when you want to retain the subjects, it's obviously good sense, social sense, that if you have a good rapport with the subjects, if they understand your protocol well, if they understand the responsibilities well, and they have an informed consent of a few documents, and for the minutes, that you will retain them, but you also have to send them kindly reminders of when their NIT visits have to be coming in, although they have every right to withdraw from the study at any time, regardless of whether they've done 90% of the protocol or 10%. And you cannot, and should not be held against them, and normal care cannot be disrupted. That's a key thing in the rights and responsibilities of investigators and investigators. So, the next concept we discussed today was really of sample size calculation. And sample size calculation, there are a few more things that the investigator needs to know. That's what the job is to override stage. But the investigator does not know the life description, full protocol is sent to the IRB. Before all that, yesterday, Professor Pale was saying that even the statistical analysis and all should be reported, but a priori, before the thing is done. So then, in that particular case, there are 3 or 4 concepts that we need to know as clinicians or clinical investigators. Number one is, when you're doing a sample size calculation, it's important that you have the studies adequately powered. Power means the ability to detect the change when it's present. So if you normally phase 1 studies usually could be powered anywhere from 60 to 80%. If you make a high one error, and phase 2 studies, phase 2 studies in general, phase 2 and phase 3 studies are powered at more than 80%, 80% to 90% standard. Sometimes you want to increase your power to 95%. So when you're calculating that, so you need to know what your power is. So any online calculator, or your SPSS, or your SATA, or your SAS, or whatever tools you're using, has all those things. For RCTs in general, you need to know the concept of the measures of association. What are the common measures of association we talked about yesterday? One is relative risk, absolute risk reduction, control event rate minus experimental rate is absolute risk reduction. Control event rate minus experimental event rate over control event rate is relative risk reduction. That's important. And then we talked about that example where I very foolishly tried to put my hand in the electrical outlet and trying to determine if I'm alive after 5 minutes or 10 minutes. So if my instantaneous hazard of dying is 90%, that's like a hazard ratio of 0.9. If I survive that particular point, and then I keep my hand at the electrical outlet for another 5 minutes, then the probability of dying is 0.9 into 0.9. Each time there's a 90% chance that if I put my hand in that electrical outlet, I will have some kind of so the probability will go to 0.81. And these things, if you keep going another 5 minutes, it will go to 0.81 into 0.9, which is 0.72. So things like that. That's how the step down and stepped up approach of the survival analysis and calculation. So in this particular thing, the power we want to keep high, 80%, 85, 90, 95%. Alpha error is usually fixed. The alpha at 0.05, which is for this outcomes. This is very big. Then my is comparator so that's the two parts on you put in the function of I1 to be there. You continue your I2 which is minus powers to be 0.2 or 0.1. Power is 0.9 and power is 0.8 and I2 or error is 0.2. So you put in those values, that's what you need to know, and you need to know the variability and the standard deviation, standard error of the mean. All that comes with the numerator. The variability of the means based on previous studies is high, then your sample size will be higher. The numerator. The key concept I want you to be very careful about is the denominator, where you put in the effect size, which is that size that you think that you make a difference, the amount of difference is clinically significant. So if I take somebody and I do a study looking at a drug which improves ejection fraction for heart failure, I might say that the ejection fraction, if everybody has roughly around 40% ejection fraction, and I want to improve that to 50, then I'm talking about an effect size of 0.20. So 40 over 50 100. So 0.20 is basically what I'm trying to use. So that 20% effect size is what we put in your denominator. But if I say I want, so that way your sample size will be relatively large, but still that doesn't really significant difference that we think will be important to patients. But if you say no, I want to increase my effect size to 50%. So the ejection fraction improves after 6 months of therapy from 40%, 30% to 60%, 80%. That is impractical. So if you do not tell that yourself that this discuss with, you tell you, oh wow, the sample size can be hard, can be much less, but then you will never achieve that. Similarly, if you say I want to use the effect size is only 1%, if the ejection fraction increased from 40 to 41%, what about that? We will give you will be able to achieve that. There you go. So this is the concept of sample evaluation for us. You use the hazard, change in hazard, whatever you think is important to measure the situation. And we also say that we have what is known as a data safety monitoring board. So when you're doing a study, data safety monitoring board behind the scenes is looking at and going to the analysis of the analyzing the data. And any significant improvement and not time when before or any significant harm when before. And both cases, it is a significant improvement, we don't have to study extra patients. And if it is significant harm, we won't study extra patients. But if you are also doing an analysis at 30% or 40% of the trial time, interim analysis, you might stop the study for fertility. You wanted to recruit about 1/3 of the patient, but not management recruit 1/3. Two times, you're not getting those patients in your population. That's the data safety monitoring board to ring the bell, tell the sponsor, tell the analysis is usually something that most data safety monitoring boards do, and when they do that, there's a price to pay for it because we are analyzing it for time, and usually you say like on there and back, and you have to adjust. We are collecting data as opposed to retrospective studies like retrospective cohort based control studies. The basic data in randomized control trials is usually much less because you have things under your control. Prospectively, you are collecting data. Even then, the subject may not show up at the required endpoints, and you may need more water. And you may then, of course, include collecting data. So for instance, if you find 10 readings and the reading number 7 is missing, you can see okay, I take the average of 9 readings and put the blood pressure reading for the mean arterial pressure in the 7th thing it was missing. Or you may say that I might take the 6th reading and carry it forward. This is known as last observation carry forward. Or you may say I will take the best case scenario, the best blood pressure, or the worst case scenario is worse. And then so you have some form of identifying how to address that, and that is usually done before the trial is done. Then the study question and hypothesis, we quickly mentioned that anytime you do a study, you have to have a hypothesis. A null hypothesis, which is what the experiment is basically desires that you are witness against yourself. You say there will be no difference in mortality between the two drugs or between the two therapies. And the alternate hypothesis, there will be a difference. So you are simply generating that, and based on that, you generate a PICO. PICO is a very important concept, PICO, that whenever you're doing a literature search or whenever you are formulating research questions, always say that in these patients or in this population, this I intervention compared with this comparator, either placebo or nothing or active control, will lead to a difference in outcome over time, PICO, over 2 years will lead to a difference in outcome of about 20% or improvement in mortality. This is how you formulate a research question, but then you have to remember the mnemonic final. F of feasibility, we have the patients, we have the resources, we have the time, we have the trained personnel, we have the money. I, is it an interesting question? So what if we do it and so what if we don't do it? And for novelty, has it not been asked before? E for ethical, is it ethical? What is not ethical, you cannot have a research study. And R, is it relevant to your area, relevant to your practice, relevant to your epidemiology, relevant to your content? And then we come to the question of randomization, and I want to start there again because I rushed to that. So, in this little bit of a review from yesterday, I hope things are a little clarified as to what was trying to convey yesterday. A little bit. 5%, 90%. So somewhere between 5 and 90%. So it's a very wide confidence. The result can be as bad as 1, it can be as good as 90. So this is how statistics and all the things and confidence into it. This is exactly how it works. So let me get quickly to the randomization side. So we looked at all of this. Okay, now I don't want to go through these, but I mentioned that there's a table here. You can have my slides and at the end, and if you want to review this, it's there in open source material. Survival analysis. Anytime you report a clinical trial these days, 80% of the time, the good top journals in the world, they want you to report survival analysis, which is what I try to demonstrate the concept. And when you draw the curve, they start looking like this. Either step down approach or step up approach, time to event or time of survival, or some key points in time. Okay. So coming back, so today we are going to take it from the randomization, try and prove the key elements of the internal validity, which is randomization, allocation concealment, blinding, and intention to keep analysis. RCT is defined by these 4 elements: randomization, allocation concealment, blinding, and intention to treat analysis. So we will see what each of them is. So in this particular study, the NAPA CKD study, they calculated the sample size of 4,300 patients using with a GFR of glomerular filtration rate between 25 to 75 and proteinuria between 200 to 5,000 milligrams in 24 hours to receive either epagliflozin 10 milligrams or placebo. And the primary outcome was a composite, a combined outcome of a sustained decline in estimated glomerular filtration rate of at least 50%, development of end-stage renal disease, and death for renal or cardiovascular. So if you look at break it up into the bits and pieces, the hypothesis is NAPA reduces GFR by 50% or incidence of ESRD, end-stage renal disease, or death from renal or cardiovascular causes. The null hypothesis is NAPA will be equal to placebo. The alternate hypothesis will be NAPA will be not equal to placebo. This is a two-sided hypothesis. Idea, we look at this quick study design. I want to now take you to the randomization. So we already reviewed the outcome measures in the study. There were some secondary outcome measures. You don't have to go into detail, but randomization. So randomization is simply a method of assigning trial treatment by chance alone. So when you're doing a vaccine study versus multiple, when you're doing a vaccine study versus placebo, to get the participant taking the informed consent that you can by chance alone get the vaccine or the placebo. by chance alone. And the randomization can be like a toss of a coin. We keep doing the toss of a coin, head, tail, head, tail, head, tail. You can do that, but then there will be some imbalance. After some time, you might find in 100 point tosses, about 60 will be heads and 40 will be tails. It can happen this imbalance. So those who are interested in participating in the trial, those who meet the inclusion and exclusion criteria, and those who consent are now randomized by chance. And the randomization can be done through random table charts, which were there in the past in my century, but in your century. Yeah, for the younger people, this is all done through central web-based randomization sequences. Random number generators are generated by computer generated sequences. So once that happens, so let's see what they did in this study. In this study, they are randomly assigned to receive dapagliflozin 10 milligrams or matching placebo in accordance with the sequestered fixed randomization schedule with the use of balanced blocks to ensure an approximate 1:1 ratio of the two regimens, and randomization was stratified according to the diagnosis of type 2 diabetes, yes or no, urine albumin to creatinine ratio less than 1,000 or more than 1,000, and the investigators used a voice response, a web-based response system to determine trial group assignments. Randomization was monitored to ensure that a minimum of 30% of the participants were included in either the population with diabetes or the population without diabetes. And minimum 10% of the trial participants had stage 2 chronic kidney disease, and participants of all trial personnel, except the members of the independent data safety monitoring boards, were unaware of the trial group assignments. The dapagliflozin and placebo were packaged identically with uniform tablet appearance, labeling, and administration schedules. In this entire thing, there are three concepts. One is the randomization, how it was done, stratified block randomization. The other one was the allocation concealment, which means that the tablets, the placebo, and the tablets were looking identical, tasting identical, weighing identical, having the same consistency. So nobody could tell. So that was done. And then the investigators and the patients did not know which group they were assigned to. This is the concept of blinding. The statistician did not know what was the data coming from. A is active or B is active. They didn't know. The outcome assessor who are looking at whether the outcomes are developing, they don't know that this group is the active treatment or this group is the active treatment. This is all the concepts that the report in human medicine is describing like that. But when you read it next time, you will see and remember, inshallah, from this workshop what it means. So the reason to randomize it, that it protects you from selection bias. You're not influencing the ability of something to, you're not influencing the selection, and known and unknown baseline characteristics, known and all the baseline characteristics are balanced, and known and unknown confounders also balanced in the two groups. So you can, so we already talked yesterday, simple randomization, permuted block, where you mix blocks of 4 or 6, 6 or 4, so that you cannot guess who's the next one in line. Stratified randomizations to balance the genders, to balance the diabetes, to balance different factors in both groups and make the genders and everything equal. Cluster, where you randomize things in clusters or practices, and adaptive randomization, where you try to tweak the randomized process to make sure that at least some of the participants have a particular outcome like diabetes. So simple randomization can lead to an imbalance, like you're seeing in treatment A and B. Permuted block can cause a balance, but imbalance again in the male and female gender. Like last slide, like this one, we can blue, not balance. And stratified randomization will give you, in the end, the males and the females, the pink and the blue are both assigned equally in treatment A and B, which is what you want to remove the effective gender. So this is the randomization that we can move to. And now going on to today's session, in the next 15 minutes, we are going to look at a few of the concepts of allocation concealment. So basically, I said you don't want anybody to be able to guess what is the next sequence of randomization. So you either use a placebo which is identical in every way possible, or you use sequentially numbered opaque sealed envelopes and put the sequence in there, A, B in there, and they should be opaque so that nobody can put them to online and try and wait or guess the sequence. People have done that in the past. It is done before allocation testing, before the randomization process, and it prevents selection bias and undue withdrawal from the study. But if you do not do it, the allocation testing properly, then you may introduce these factors. So people have actually done this. In the next slide, which I'll show you this. They have actually put this up. So when we look at the results, when they come down, if they're happy, we know that we have failed. They give you another set of registration forms. If they like, you know, we have passed. Even that is the breaking of the sequence that you get. And you don't want anything. So in this data, the patient says the patient's condition fits the trial and she is consented. It's not fair, it's not fair. The patient is eligible. 22. After that, I will tell you what we do. This is the way this study is done. There is no way. Masking and blinding, everybody at different levels is unaware. Either it is an open label study where both limbs, both investigator and the patient knows which limbs you've been assigned to. Sometimes you have to do that, especially in surgical patients, surgical trials, or single blind, maybe just the patient is blinded to it, investigator is not blind. Or double blind, both investigator and patient are blinded, they do not know what the treatment assignment is. Or triple blind, where patient, investigator, outcome assessor, or statistician. So different levels of blinding, you can bias. And if you do not blind, you will have problems with patient response bias and patient bias by the investigator's evaluation or classification of response and events will be unduly detected in a group which has active treatment because of your subconscious bias. So if you do not know, if people knew that they have the intervention, they will be smiling and walking better. And if they want to see, they will be saying, oh, I'm getting this. So they will be very happy. Tell them that you will not know how they are. And you later on you can deliver a questionnaire to see whether this blinding is successful. But sometimes when it is a major safety issue, then you have to break the blind and tell the investigators that this was the limb A, this was the limb B, because lots of people are having side effects of concern in one limb. So allocation concealment occurs before randomization and blinding occurs after randomization. And when you look at this, this is intention to treat analysis that whenever patients are randomized, they will go to treatment A or treatment B, but some in treatment A will not follow that treatment. And some will follow that treatment. The treatment B, some will follow that treatment, some will not follow that treatment. The whole idea is that if you are randomized, even if patients drop out or cross over to the other limb, you still analyze them as if they completed the study, the same limb. This is known as intention to treat analysis. And if you do the other one, which is as performed, whatever drug they actually took, which is known as protocol analysis. So this is these are the four key components of our RCT. So the sample size calculation in the DAPA CKD was done in a particular way. The power was 90%, the alpha or was 0.05. They had proportion. It was as randomized was analyzed. That is one thing you need to look at all these studies, and that is how you report. Okay, now I'm starting. Anyone can help you? Can you move slide? On fixed now. So these are and the hazard ratio, confidence interval, all what is the cost proportional hazard is simply a proportional contribution of several different aspects of the relevance of an outcome that you compare in a survival analysis. This is the result that the red one is the placebo, the blue one is the nitroglycerin. Every layer, the hazard ratio is less than less than 1, which is the point of contact. 0.61 means 39% reduction in this in the nitroglycerin in terms of cumulative incidence of the primary composite outcome. And renal specific outcome also the nitroglycerin was better. And the death from cardiovascular causes also that the nitroglycerin hazard ratio was 0.7. The confidence interval was 0.55 to 0.92, which means that either you would have as wide difference as 0.5% reduction in the hazard or 8% reduction. And that is the main for statisticians and clinicians to be aware of. This is a subgroup analysis, what is called a forest plot, and in every subgroup they showed that the nitroglycerin was better than placebo, except when the trials were conducted in Asia. And in Asia, although in Asian population it was good, but when we did studies in Asia, because we did not learn the techniques of randomization and randomized controlled trials, we made mistakes in Asia in this particular study, so our results were not significant because they touched the mind, the confidence interval touched the point of no return. So this was a epically done study. Basically, there was no real reason, nothing new in nephrology in terms of improvement of outcomes after ACE inhibitors, and they use this in addition to ACE inhibitors to change outcomes in both diabetics and non-diabetics. And the study sponsor, the pharmaceutical sponsor had no influence in reporting or outcome data. So one of the statements that one should be aware of is what is known as the Comstock statement. This, if you look at your handout on Google, I've given that. This is how you report an RCT control chart, who's randomized, who was allocated, how many were lost to follow-up, and how many are analyzed. And this is the table which gives you all the components that we've been discussing in the last 2 weeks. All the components are there. So when you put this in front of you and design a study, or when you are writing up the study, use the Comstock statement, and inshallah it will be accepted as the best of the results. So I will skip the applicability issue, but I'll just mention one thing, that in this particular study, everything was nicely done and very well blinded and stratified and concealed and randomized accordingly. The baseline characteristics showed both of them were equally balanced, so that whatever the intervention was the change in outcome. And the results, once the results are there, you look at this control event rate and the experimental event rate that we talked about and the confidence intervals. But this is the key I want you to concentrate on. So you have a relative risk of a hazard ratio of 0.61. The absolute risk reduction was 19 minus 14, which was 5.2%, 18% and 14%. The number needed to treat is 100 divided by 5.2, which is 19, which means that we needed to treat 19 patients in order to change the outcome in 1. And that's not bad if you're talking about changing outcome in 2 years for kidney disease and putting people on dialysis. So that is the way we analyze this, and you see if your data is applicable to your patients. A clinical investigation center, you have everything in here. All you need is investigational medicine products for storage area. You have a lab, you have a data management center, you have an IRB, you have a trial subject assessment area, you have a radiology, nursing, you have pharmacy, you have beds, you have the ethics. IRB, all you need to do is get some standard storage equipment for investigational drugs and get your body to approve you, and you'll be fine. This is our legacy. We celebrate James Bond every year, May 20th, for doing randomized controlled trials, but no, even if we did that, there was a fragmentation 1,000 years ago, 1,200 years ago, and Nazi talked about the concept of control groups. So I acknowledge all my team members. All this presentation is a combined presentation of my team members who were there with me in the comstech first randomized controlled trial. I've combined their slides, combined the things with their all my mentors and colleagues and comstech and of course, I say Asante and Billy, Wish keep hosting us. Let's go to the quiz. We have 7 minutes to do the quiz. 6 minutes. Can you go to the quiz?
Thank you so much.
We are ready.
5 minutes. Okay.
Let's just do the quiz. Yeah, the second one. Okay. So
Do we give chance to people if they want to answer?
let's go to the first slide. Give you the slide view. Yeah. Okay. So it's a self-assessment quiz, and there are some prizes for some key answers. For the simple answers, no prize, but even I don't know which one will get prize. Only Allah Subhanahu. It will be randomly given. Okay. Usually a data set board, I talking about that, and let's go to B. This technique, it shows proportional representation in a trial. What's the answer? Okay. Stratify. Stratify randomization. Yes, that's right. Remember, I already defined Pakistan. So we are right. So we take it out any of the word and please give it as a gift. And I found some sweets here as well for small business. Or here, or in my pocket. For example, the other thing to Dubai, inshallah. And with your permission, with your consent. And so let's. What is it? Please read it. It has something of maybe of value 15 minutes. Okay. Next one. This is the most important of the measure that the study seeks to answer. Lots of stakeholders give them all these views. And give them one book that they give us answer is going to write you. And then who answered first? Ask your conscience. Okay. This is the genuine clinical answer principle amongst practitioners regarding the best healthy patients in performance RCTs. What is this? You can only do RCTs if this principle is satisfied. And if you give a wrong answer, I will take it. And then I'll connect it right answer. So which one? I don't know. Say I don't know. I don't know how to use it. This phase of clinical testing assesses both safety and Yes. And time to event or survival as accumulated probability is known as? Excellent. Same, we got the prize several times. The integrity and strength of the study design based on all these things is known as? Randomization, allocation, blinding, and intention to kill is known as? Internal validity. Yes, all of you. Good, good. Shabash. Wonderful. If a study has a broad inclusion and narrow exclusion criteria, that will be associated with external validity. Give her a speak, please. Yeah, the book I can give you online. How you come to Pakistan, inshallah, my wife can give you 700 books there, like in the story. These studies are typically conducted in 25 to 100 healthy volunteers to determine drug safety. Phase one. You got a prize already? Yeah. Okay, so I'm not going to take any more time. The quiz had a few other concepts that we could have done, but I am going to give a special prize for this. 1 divided by absolute risk reduction is the last question, which reflects the minimum number of patients required to be treated to show benefit in one subject. Yes, ma'am. What? You already got a prize, right? No? So this was the last one. And believe me, I'm in my senses. There is no, you get 100 dirhams. So that's the end. Dr. Ahmad is going to
Thank you, sir. No, you forgot to give me. All right, the next presentation is now Dr. Ahmed.
Okay. There's and go again. Okay, you get those things. So you should get
any trial protocol. Anybody? Can you ask the question again? Okay. Have you seen any protocol? This is Dr. Mas. Yes, I have. Okay. Okay. So my next question is, why do you feel? What do you have a thinking of why we actually need a protocol? Or
in simple terms, why we need a proposal.
Thank you. All the potential flow ones are going to move so that you can cut out your side, TKP and it.
Okay. So, correct.
This was why you want that's the reason I was pushing. That's pushing the also that's how we're going to be.
Okay, great, brilliant. So, a protocol is, all the answers are correct, but in my slides, what I'm now going to present is, there are many more things which we should consider to see a protocol. Right. Okay, so I have a great idea, strategy. Do I have appropriate patient population? I do have such a faculty, disciplinary people around, and supporting clinical staff, and the time and administration. Then I have funding and all the legal and contractual issues. So I have to figure out all these things before developing a protocol. So in my slide, you can see I have written something in green. This is why I'm telling because that's how to have a particular meaning. To predict and make allowances for the real world behavior of different individuals who will be involved in the clinical trial, with the expectation that they will behave according to the protocol. So when I say behave, that's something we have to recognize that a trial is an in-depth investigation of comparison of certain investigator of investigative medicine. So therefore, lots of people will be involved in it, like a clinician, a pharmacist, a nurse, and then there'll be people from the legal aspect, then there'll be people from the budgetary aspect, then there will be people like statisticians, then there will be an IRB, then there will be people from the regulatory aspect. So you can appreciate that there are lots of people with this link to that to any particular protocol. So if you have a roadmap of such a document, where you know that everybody has a different understanding of what they might have, but you also want that they should follow a particular path and they should not deviate. You had to write out certain things so that everybody should follow that, and there'll be no difference of conducting it. So the protocol will potentially be used by many different people, as I say, including data managers, statisticians, proprietors, ethics committee. Everybody will be seeing it from their own perspective. So technically, in simple words, it's a document that describes the objective, design, methodology, statistical considerations. Usually it also gives the background and rationale for the trial. So you might have heard research proposal. So it's often used technically, a clinical research proposal is a rather brief document which describes an idea, while a research protocol follows a proposal stage and serves as a detailed roadmap and report detailing how you plan to complete your project. Okay, so this is just the difference. You might have heard of a grant proposal, and I'm also telling about a clinical trial protocol, and I just said in my slide that it's like a clinical proposal. So you can just appreciate some differences. Grant proposal is a scientific document, while a clinical trial proposal protocol is a regulatory. Then grant proposal is actually written for getting grants, while the protocol is not intended for getting any money because the money is already secured there. So it describes single study in detail. Then program proposal, the focus is on aims, hypothesis, and methods. While in a clinical trial protocol, as Dr. Kali has briefed in his last talk, that we were focusing on endpoints and specific objectives. Then we have a grant proposal which talks about cost, staffing, and equipment. While in clinical trial protocol, as I said, that we are more focused on how we are going to implement this stuff. So you see, the mandate for both of these documents is different. Then the grant proposal is reviewed by scientific committees, review committees, funders. While the clinical trial protocol is actually developed by the sponsor, and it's reviewed by scientists and IRB. Plus, then the tone, the way it's written, is like grant proposal is the tone of the grant proposal is persuasive because we're trying to get funds. While in clinical trial protocol, we actually try to put as much detail as we can so that we cannot leave anything behind in order to implement that study effectively. Okay, so how to write a protocol? Now I have said that as we have to write a protocol, we have to provide a roadmap. And to provide a roadmap, I must have an imagination of how things will be going, what will be coming before, and what will be coming after. So it's a matter of judgment. Rule of thumb is include the right amount of detail necessary for the reader, for each section to be able to understand exactly what is required to conduct the study. Now, how can the results be reproducible? So I think how many of you people have actually watched a cooking recipe? Any video or have read through any cooking recipe? Have anybody of you have gone through any cooking recipe? Okay. So for a good recipe, the people who writes, they actually wants that whoever reads that particular recipe and reproduces exactly what's intended to be in that recipe. So in a similar fashion, whatever the detail we try to put in a protocol, we actually are intending that whoever reads it. Now, by saying whoever, I mean that protocol will be read by nurse, by a pharmacist, by a principal investigator who is usually a clinician, or by a physician. So anybody who has, or there might be people around who, as I said yesterday, that they might say, oh, I am not satisfied with the result and I want to redo this system. So they might read that protocol and they want to reproduce the results in order to check its authenticity. So that protocol has to be that extent. So we start how mapping out the study schedule. So we have a drug and we want to develop a protocol. And now we have to consider some events so that we can have an idea how should we initiate writing a protocol. So as you can see, that there is an element of informed consent. So we are trying to say, okay, when the informed consent will be taken, usually it's always taken at the start of the study. So so that after that, it will proceed. So it will be informed consent, there will be check about inclusion criteria, special medical history, primary diagnosis, clinical assessment. So you can expect we are trying to enlist all the events which are going to take place while in a particular study. This is how we are judging things. Then there will be randomization. Probably randomization can be on the same day or any other day. That will be and since the patient has now been enrolled and is given, so there might be adverse events, and there'll be assessment of relapse. So it's just you can just say it's just an idea how we are going to map those events. So as Dr. Khalid's lecture yesterday has mentioned, a protocol can be published, not the study. A typical trial protocol can be published before the study starts. So I'm going to encourage you to read this particular protocol. This was a study conducted by McMaster, University of Canada. I had a chance of contributing to this particular study. This was the study. It was the investigator initiated study. What happened in the study was that there was a question that whether reduction in time can improve outcomes of hip fracture patients. Like typically when a hip fracture occurs, the hip fracture surgery takes place around after the time of the decision, it takes about 48 hours afterwards, after which the hip fracture surgery takes place. So some of the clinicians thought that can we reduce this time of surgery and this might improve certain outcomes, because at times this happens if the person goes with a hip fracture surgery. And after you can say 20 days or 30 days, there are the people who get pulmonary embolism or some other non-embolic events. So the clinicians, while considering this research, thought that can we improve the outcomes by reducing that particular time. So they were thinking that if we can do a surgery within 6 hours of the time of the patient versus the routine limb. So the routine limb was the same 48 hours. Why? The new limb was within 6 hours. So you see, this in this particular trial, no drug was involved. It was just a trial where two different methods of how we're going to treat patients was followed. And I said that there are certain studies which are actually done that to improve clinical practice. So this particular study was exactly like that. I'm going to encourage you to read this particular protocol. So in this particular protocol, they have actually enlisted a table, but they have actually shown that how the study will take place. So right from the beginning, they're showing participant screening, and then there'll be participant randomization, then there'll be an accelerated care group, then there'll be a control group. In an accelerated control group, what we'll be looking for. And then both of these limbs will be followed. Then there'll be analysis of intervention, and then there'll be distribution. So you can see they have actually provided a roadmap of how the sequences are going to take place. And they've actually mentioned this in the trial protocol. So in a typical trial protocol, we have a list, as I say, you need to have a recipe of certain elements of how to write protocol. So there's a title page with some contents, then the email how we are going to report treatment regimen, then will be table of contents. Afterwards will be objectives. You must have to mention it. Are there any primary endpoints, are secondary, tertiary? That's there is a background. We have to provide a rationale of why we are going to do that study. That background has to be very strong. We are going to talk about the inclusion criteria, then treatment plan, and study design. So you can ask that is there a standard checklist of what particular elements are typically included in a trial protocol. So there's a statement called spirit statement. So this spirit statement is widely recognized, and it provides a very exhaustive checklist of all the elements which are to be in a particular trial protocol. So this is a very exhaustive list. I also encourage you just go through this list. I must say that in a particular trial, there might be not all the elements fit into it, but lots of elements will be still there. So for writing a plan protocol, if I want to start, and I already have a checklist, that will give me a very good idea of, okay, this is what I have to write, this is the section I have to fill, this is the section I have to fill, because and they also provide how you have to fill. This is statement can be really helpful. Okay, so there was a part of disorganization. So I just picked a section of the hepatitis protocol of ethics and dissemination policy. They have actually followed the spirit statement with reference to even they followed the spirit statement for putting every right ingredient in the trial protocol as per spirit statement recommendation. Okay, now comes any question regarding a trial protocol? I know it's rather, you can say, documented. I think still you have any questions?
Thank you, Professor. Just one question, to guide. We talked about publishing the protocol before the period study. My boss was that one defeated as a faculty. For example, I'm doing a study and I published the protocol. Later on, I find my findings. Remember, it's part of the protocol, also named part of the publication of the final study. Was that one defeated as faculty? Thank you.
Okay, so actually, it's worldwide it's encouraged that we publish the trial protocol, but since it is doing this transparency, but it's not mandatory that we should publish it. Most of the people actually don't do it, but still encouraged. You can see you'll find and on practical terms, there are two. You can say there are two groups of people. This is just my, you can say my position of understanding. Lots of trials, particularly drug trials, since there are confidential drug trials, their protocols are not published. But on the other hand, our main core academia, they actually are more interested and more focused on keeping a very transparent way of putting things in front of the public. So they always encourage publish it beforehand. So if somebody has published a trial protocol, the matters of confidentiality and things like nobody else can do that, it doesn't usually make a big difference. Or say, for example, we have a research person and two different people are working on the same research push and it can happen. Still, I don't have seen a fight on it particularly. This even happened in COVID, that a person was working on a research question and a person from another country was working on the same research question. Still, they produce, and even they produce different findings. One person produced another finding and the other produced on the same research, produced some other finding. And both were published. And both were having cited as well. So actually, in a scientific community, this is actually considered as the beauty of publishing results in a transparent way. Any other questions?
Yes, thank you. Just to respond to this concept, I think usually what is done, you cite the protocol in your ultimate finding of the study. So when you're writing a manuscript, you cite the protocol in the manuscript. And by experience, what I've seen is usually when you try to publish the protocol, it's set out of the area within which you had the study approved. So you get more experts to look at what you're going to do. And sometimes they give you very important suggestions or things to change that may require you to actually go back and modify a few things to the ethics committee. So that becomes very helpful to the study in general.
Thank you. A very brilliant contribution. That's something really good to have. Is there another one? Yeah.
Just one practical question. Is it possible to publish a protocol as a living document, or when you make an amendment at the problem, how do you deal with this?
Okay. So you write it as it is. This might be a possibility that you develop a protocol and you started working on it and then you realize I need to amend it. And this in practical terms always in most of the trials that the protocol is amended. So in clinical trial world, this routinely happens that there's these are the things like protocol version number 1. Then after the initiation of the trial, something were amended to the reference to inclusion of participants or statistical analysis or any other thing. And there's a version number 2 coming on, and there's a version number 3 coming on. So this happens. And usually all these things, and because at times you, as I said before, we are all on the path of the journey of learning the right truth, but we do not have it. So, and in practical terms, so when you start certain things, then you realize, okay, I hadn't included this, but this has to be included. Or as our good fellow suggested, that some experts might have some really suggestions that can improve that particular protocol. And and therefore a new version can come, and then that's included, and that's how the plan protocol is executed.
And the journal for this, it's already published in a journal.
I actually didn't get your question.
I can say it gets make an amendment. The protocol is already published in a journal. Like for instance.
Okay,
yes.
so we address that in a way that we actually mentioned that this is what the trial protocol was, but when the actual trial is published, they actually mentioned that this was amended so many times, and this is how the versions were released. There can be another way, but I hadn't had a chance of looking towards that. Is when you publish a protocol and you amend it, I don't know. People don't give an errata on that because it's not an error. It's an intentional change to improve or the trial conduct. So, but whatever the changes are made, those changes are then addressed in the final trial publication. They mention it in a paragraph that we have amended it so many times. Okay, so moving on to our next part. Okay, so how many of you have actually gone to shopping? And, I hope you people all have moved to the market. So what I'm asking is, you all make budgets before going to shopping. You must have an idea of that this is the amount I'm going to spend, and this is how I'm going to spend. This is an area I'm going to spend on clothing, and this is how I'm going to spend on shoes and whatever commodities I'm going to use. You have an idea of what I'm going to spend on. So, clinical trial protocol is a very expensive experimentation and interventional study. So, we need to have a defined budget as well here. So, as I said before, when you go to shopping, you know what you're going to spend on. In a similar way, you have to understand the protocol, the required procedures and scope of work. Then is this clinical trial feasible with the previous clinical research program? Are the requirements of the clinical trial reasonable? Is the patient population available? So all these questions we ask ourselves, the group of investigators, all they ask these questions. Our conclusion criteria to know broad as Dr. Kambi has briefed in his presentation. Okay. So, is there appropriate study staff available in the study? So we need to, before costing things, we need to see if we have all the things or not. So this is what happens usually while imagining about budget. Like we're taking impatient when we're designing a budget, and we are even more impatient after we have designed the budget. So what's the rationale? It's the same thing. We should know firsthand what amount we need to spend on in that particular study. Then identify who to pay what. There are certain hidden costs which I'm going to elaborate in my next presentation. Then there will be documentation, of course, because the trial required lots and lots of documentation, lots and lots of documentation. Then there'll be to identify research versus standard of care. Okay. Anybody have an idea of why I mentioned this research related versus standard of care? It has a very strong implication with reference to budget. That's why I'm asking this particular question. Anybody have an imagination of research related standard of care? To elaborate this, I'll give you an example. So now I have mentioned a trial protocol, the HIP attack. So this refers to budget. So for example, my comparative link was that the routine care procedure is like there'll be a person who is impacted with that, and after 48 hours, that person will receive the surgery. Okay, fine. Those 48 hours is spent in a way that that person is has routine tests, gone through certain tests. Then there will be a anesthesia fitness. If that person is having any other comorbidity, that is taken care of. And then afterwards, the anesthesia department, if that person is a fitness, that person goes for the training. So now I'm asking that I am now trying to compare this with our research where I'm imagining that I have to conduct that surgery within 6 hours. So I have to fast pace all my departments. I have to ask my anesthesia people to fast track that patient fitness. So there might be a possibility that lots of patients might exclude from my inclusion-exclusion criteria. But even though some patients still can get, the patient says yes, some people can go through within 6 hours surgery. So for those people, I have to now fast pace all the procedures that requires expenses. So that particular expense has to be taken into account. Okay, so industry sponsored versus grants, subject to different sets of regulations. Okay, these are certain technical terms. I must elaborate this. So in terms of budgets development, if a trial is funded by a commercial company and the intention is so ultimately this particular trial is intended for a particular commercial use, the costing, the way it will be costed will be different from the studies which are actually investigative initiated. Because most of the investigative initiative studies are not intended for any commercial aspect. Therefore, the way they are being costed is different. Industry-sponsored projects are not capped. By capping, I want to say that we put a limit on certain figure for investigative initiative, but for industry, we don't put a cap. We can inflate that figure considering the needs of the trial, considering the needs of the institution. Like, let's say, for example, in my routine consideration of developing a budget, there is an indirect cost involved while making a budget. That indirect cost counts for the infrastructure the institution is providing, and for that indirect cost, usually the institution wants to get some money in order to maintain that infrastructure so that that particular study can be conducted. For usual investigative initiative study, usually the indirect cost is around between 8% to 15%. Most of the sponsors, which are usually government regulatory authorities or government agencies, don't even provide to give an indirect so much indirect cost. So they wanted, okay, you just want to stay within that particular 8% to 12% cost. In fact, the NIH USA, when dealing with any international or other organization outside USA, they had put a cap of 8%. But with reference to drug trials, if you were dealing with drug trials, you'll appreciate that the indirect cost they provide you is more than 20%. So this is something of the whole budget. So for industry sponsored trial, you have more freedom to negotiate. Not typically take it or leave it, but if you're dealing with some government agencies or demand agencies, this is what they have. Either you take it or you leave it. So most of the institutions have different policies. So you look into what needs to be covered. But it's okay to do a little better than just cover the costs. And this is what we do. We respond and we investigate initiative study. So there are certain other things which needs to be considered, like the difficult and complexity of the study preparation requirements, patient number, how long the study will take place. Why I have mentioned how long the study will take place, because the length of study also determines cost. The longer the study will be, the longer will be the cost. Like most of the oncology drug trials are 2 years or 3 years. We just don't cost the interventions or the treatment arm. We are usually following patients for 2 or 3 years. So for oncology studies, an example, follow-up required lots of spending. Like if I have done a trial on an oncology drug, now I need to check whether that particular drug or whatever I'm comparing it with was whether working or not. So I need to have CT scans, MRI, blood work, and if I am following those patients for every month or every 3 months for 3 years, just try to imagine the cost I'll be having. So drug trials do provide set amount of money because they want to prove it that yes, whether the drug will be safe or not. But this is how the cost imagination comes. Then there'll be inclusion exclusion criteria for subject improvement. And then the staffing, how big our team will be. Usually it's very, very tightly calculated how the staff will be there. Then what to consider in a clinical trial budget, and there will be invoiceable costs. Certain investigations. In fact, most of the investigations are charged at most of the systems as per actual. But in trials, you can inflate a bit. Then there will be hiring fees, protocol amendment, adverse event certificate, adverse event management. So, say for example, if somehow a patient has taken an investigative medicine and he develops some sort of reaction or anything, and he needs to be taken care of by the hospital again, where that cost will come. So that is also covered in the trial, and that is also provided by them. Then there'll be safety reports, then there'll be audits, time and efforts of study. So auditing is coming and going. So all that is included in the budget. Then there'll be screen pages. You screen for certain patients, but they were not able to be enrolled in the time. But there was a cost incurred in it, so you need to incorporate that as well. Then there will be shipping of samples, packaging of. So for particularly for infectious disease trials, there is a very, very hectic and a very tight packaging of those things. And that packaging or the cost of that packaging is provided by this. Then there'll be study staff time, the monitor. So the time the staff is providing, that is also incorporated. So, okay. So one thing I am showing this particular table now again, but with a different mindset. Now, if I am making a budget, I see this roadmap in a different way. So say for example, I'm seeing there'll be participants screening and improvement. So if I have to see what I'm not costing, so there'll be an expense of information of a fracture. So I'll be costing, okay, I have to do an X-ray for those particular patients or any other superior investigation like a CT scan to confirm, okay, so that cost will be there. Plus, there will be eligibility assessment and other blood work involved in it. Then there will be possible randomization. So for that randomization, I need to have a computer software that will be randomized, and so I have to purchase that software. So the cost of that software I have to keep in my mind. Then there will be accelerated peer group. So one thing I'm bound to mention, and this principle I apply it very frequently. In a particular clinical trial, we try to cost the intervention, not the standard of care, the standard care. So in a particular hospital, the patient is getting standard of care as the person is getting. But if an intervention has been done, the cost of that particular instance should not be transferred to that particular thing. In fact, for a particular trial, usually what we do is, if a patient is enrolled in a trial, we take care of all those particular investigations. In most of the trial, the list of investigations actually ranks. There are more investigations which are required, but say for example, certain tests are routinely done, regardless of whether the patient is enrolled in a trial or not. So for lots of studies, those tests are not included in the cost in a particular trial. Then there will be a follow. So now I consider that I'll be having a telephone bill as well, which I have to pay. So I have to put a cost in that. Then there will be analysis of prevention. So I need to pay by citations as well. Then there will be distinguishment results. So I need to consider, I have to put a publication cost as well. And then if I'm putting results in it.
What about regulatory share? What about regulatory share?
Okay, definitely. That is coming in. So there'll be data management expenses, study related personal costs, equipment, diagnostic fees, publication costs, and then there'll be startup costs. There'll be cost for site selection visits, site initiation visits, off-site meetings. They all are taking care of the cost. Plus, as I said before, because I've shown you a roadmap, there'll be first and so there will be initial enrollment costs, subject to screening, in 10 years. And things will be charged, group charges, hospital bed, film duplication, where drug will be stored, who will be paying for that. There will be lab work, radiology, cardiology, pharmacy dispensing, and so everything is accounted for in a file. So, and there will be recurring costs as well, which will be coming again and again, like investigative fee, study coordinator fee, subject to stipend. So one-time cost consideration. So this is IIB preparation, then advertising, document translation, publication fee, closeout fee, and definitely as Dr. Abdul suggested, there will be a lot of there's money regulatory fee also there in this one-time cost consideration. So as I said before, research-related expense versus standard of care. Identify procedures and tests that are research and standard of care and clearly note them in the budget. So this is a snapshot of a typical trial budget. So you can see that there will be a screening visit, then there will be a baseline visit, that will be visit 1, 2, 3, visit 4, then there will be final study visit. And whatever procedures are happening will be costing as per patient visits. This is how they're, so these on the one side, we have all the events which are taking place. Okay, so you have an idea of how this is how a typical trial budget. Usually, trial budget is kept confidential, but this is how it looks like. Most of the trial budgets are generally put on a generic template. Okay, so there are some common budgetary mistakes like underestimating the time required for startup, site initiation. Then there will be exclusion criteria we do not account for. So in clinical research, it always takes longer than you plan. So you have to understand its financial implication. Okay, so just line one, if you have not documented anything in the budget, most probably you're not going to be paid for it. Remember, everything is an expense. So we can say that our question, we are going to have question and answer session around 3:30. We are going to have it for like 30 minutes and other discussion. Please, whatever you have, please just question. You have just time. So we want to invite the next speaker who is online from the University of Granada, Spain, Professor Khalid Said Khan. So he's going to talk to us right now on systematic review of a randomized control trial. So, Professor Khalid, are you there, sir? Hello, Professor Khalid.
Are you able to hear me?
Yes, we can.
Okay,
can go on.
so if you don't mind, I apologize first for the little interruption, but I can start now if you agree.
You can, sir. Go ahead, sir.
Okay, thank you very much. So today is the second day of your workshop, and my task today is to talk about systematic review and meta-analysis of randomized controlled trials. Just before we progress, I can tell you that I wrote a book on systematic reviews, which has been awarded the BMA Medical Book Award, and its third edition has only been released just in December last year. And my presentation is largely based on the 5 steps summarized in a summarized form that are described in detail in the book that I just showed you. So in the presentation today, I'll give a small introduction, then give you some detailed presentation of these 5 steps. And then I'm going to give you some writing tips before reaching the conclusion. I should highlight that meta-analysis, the term, refers specifically to statistical synthesis, which is step number 4. And the systematic review is a combination of all the 5 steps listed here on the left-hand side of your screen. The typical issue concerning any paper, including a paper based on systematic reviews, is that we often have to go through a long cycle of revision and resubmission before acceptance. But I hope with today's presentation, you'll be able to make a strong manuscript that may be accepted in the first go on the first submission. So instead of boring you through my own curriculum that I provided yesterday, again, I will just jump directly to what are systematic reviews and how to avoid confusion about them. So primarily in the published literature, there are a very large number of things called reviews. They may include even editorials and commentaries, but they are not necessarily scientific articles. They are subjective opinions expressed by experts. A systematic review is a subset of these articles. And here the key feature is that a scientific strategy is used to avoid or limit the possibility of bias in in the putting together of the evidence. Another word used is evidence synthesis. Now, some of these systematic reviews will benefit from performing the statistical synthesis called meta-analysis, but meta-analysis is not an obligatory part of a systematic review. In fact, in some circumstances, it would be actively discouraged because the evidence will not be suitable for performing a statistical synthesis. So with this definition clarified, can I just check that you heard me clearly and that I can continue?
Yes. Well.
Okay, thank you very much. So we now move to something that I mentioned yesterday, which is that research translation goes through various stages. And you can see here that evidence synthesis and guideline implementation are known as T4 translation. And this is how what you are learning about with respect to trials fits with the evolution of the evidence generated into a product that can eventually impact healthcare. And that impact is achieved or should be achieved through a scientific exercise that puts together all the trials conducted on a particular question through a systematic review. So, as I mentioned yesterday, the objective of conducting trials isn't just to produce a couple of lines in your CVs. It really is to improve the outcome for patients, communities, and society. So if you think about it, just 3 years ago, in this very month, in March, many countries underwent a quarantine to deal with the problem of coronavirus. And today, those restrictions have virtually all disappeared within a period of 36 months. And this has happened through the conduct of trials of vaccine and other trials necessary to control the pandemic, and summarization of the evidence concerning these intervention studies in systematic reviews. So the pilot and feasibility studies can be put together systematic reviews. From this, we learn how to do more of these studies better and inform how to do the large multi-center studies. And it is then putting together of the large multi-center studies in systematic reviews that we are able to achieve health impact. So your main focus is to learn about clinical trials. So here I present an example of a trial that is conceived in 1996. It recruited its last patient in 2006 and is published in 2009. So you can see that a clinical trial has a relatively long time span depending on the question and the number of patients and the duration of follow-up required. But in the life course of this particular trial, at the time of conceiving the idea, a systematic review was carried out. And here you can see a publication, title page of the publication related to this review. You've been listening to the construction of a budget. Presumably you create the budget to make a grant application. I can bet you that a grant application is unlikely to be successful for a clinical trial unless the application includes within it a systematic review of the topic within which you will carry out the clinical trial. And this systematic review should be used actively to help design the clinical trial for which you are seeking funding. So this first review that highlighted here in 97 was one of those reviews that underpinned the grant application for funding of this trial. Then during the course of the review, another systematic review appeared because other parties, apart from the researchers involved in this trial, are undertaking research in the same area. By the time the trial is to be completed, yet another systematic review appears, an update of the previous review. After completion of recruitment, but before publication of this trial, yet another systematic review is published. On publication of the trial, a further systematic review is published, this time with a more sophisticated approach to meta-analysis using the raw data of all the trials on this topic. So you can now see that the life of a clinical trial is integrally related to systematic reviews and meta-analysis of the topic concerning which we are undertaking our clinical trial. Now, systematic reviews, I mentioned to you earlier, are important for implementation. Here is an example. This is Scotland buying more than 150 kilograms of albumin a year, every 6 months, according to this this timeline, in order to treat people who are critically ill. A meta-analysis then shows that, in fact, using albumin is harmful, increases, not decrease mortality. And it's only after this meta-analysis that combines these various randomized trials on the topic that practice in fact changes, and the amount of kilograms of albumin purchased in Scotland to treat critically ill people in fact comes down substantially, because it's now learned that this intervention is in fact harmful. So we now return to the 5 steps of systematic reviews. The first step is framing the question. With respect to the clinical process, we go through all the different phases of starting from understanding etiology to making diagnosis. It's important to understand that clinical trials typically relate to therapy research, and therapy research attempts to create interventions or evaluate interventions that will improve the prognosis of patients. So we can undertake trials by randomizing individual patients, but we can also undertake systematic reviews of trials, combining the data existing in published primary studies. So this is the structure we would consider using for framing the research question concerning systematic review. This will be virtually identical to the way you will frame question for a trial, except here the study design will be systematic review that will put together data from primary studies. Again, a repetition of how a trial question is composed. Primarily, you combine various studies that compare a treatment with an alternative intervention to assess the outcome of patients exposed to a treatment, to a new treatment or the alternative. I highlight here reporting of the extent to which patients and public have been involved in research. This reporting guideline is called GRIP2. I highlight this here for you because with respect to framing outcomes in particular, it is important that the outcomes selected for systematic review focus on what is really important for patients, not just what is important for computing a small sample size that is manageable, but it should be an outcome that will change the future of or the prognosis of a sufferer of the condition for which the intervention is being developed. And here collaboration with patients and the public can be highly relevant, and therefore citizens can have a part in planning and conduct and dissemination of the systematic review. And here is an example of how journals are requiring the reporting of patient and public involvement in papers. So we covered yesterday, and I wish not to repeat how the study is designed, but I'd like to show you a little bit about how data are taken from trials to undertake some calculations for performing meta-analysis. So as we know by now, patients are allocated by randomization to control or intervention, followed up for their outcomes, and then effect size is calculated. So imagine if 200 people are randomized, and imagine that no patient or data loss occurs in a hypothetical trial. Then take it further. It's a trial concerning infertility. In the control group, 10 couples become pregnant out of 100. And in the intervention group, 25 couples become pregnant out of 100 on follow-up. Then how will we calculate the effect size for meta-analysis? So the first thing is to compute this 2x2 table where the outcome, in this case, pregnancy or failure to become pregnant, is tabulated for intervention and control. And these data appear like this. And from these numbers that we already have, you can see that it is possible to compute the rate, risk, or proportion of being pregnant under intervention or control. And the relative risk is simply a division of one rate by the other rate, and this will be 2.5. The odds ratio, on the other hand, is the computation of the odds per group, and the odds ratio is the division of the odds in one group by the other, which is that. So these calculations, as you can see, are fairly simple. However, these calculations need to, as we discussed yesterday, will apply to the study sample within each trial. The beauty of a systematic review is that because we have several trials put together, they increase the possibility that the effect sizes, when combined, become more generalizable to the eligible or the general population who did not take part in the trials. So meta-analysis not only help with improving the reliability of the effect size, it also improves the applicability or generalizability of the findings concerning a particular question. Now, you may have already covered, and it's important to know the difference between efficacy and effectiveness. In general, an effectiveness trial is what we are looking for. We are looking for how beneficial is the intervention under usual circumstances. And here, the key features that define effectiveness relate to how the sample size is recruited. For example, the people come from less stringent eligibility criteria. inside the study sample. The health outcomes are relevant to clinical conditions, relevant to patients, not necessarily simply to achieve a small sample size. The duration of follow-up is sufficiently long, sample size is big, and the analysis is by intention to treat. And I take it, and I'm confident that other colleagues will have taken you through all these issues with respect to the designing of a trial. But given that a meta-analysis combines different trials, it has the possibility to generate a summary result that perhaps will be more generalizable than individual result. Here we take an example of an infertility condition where the treatment is compared to control. Here are the data from various trials. The relative risks are plotted on this graph. Here you have the studies. Here you have the studies sorted by year of publication, the extracted data. The line of no effect is the relative risk value of 1. The calculation of relative risk we have already seen. The point estimate is usually the box or circle in the center of the data presented. Here you can see one of those calculations highlighted for you. If this point is below the value 1, in this case, it means that under treatment, less women are able to become pregnant. So the treatment is harmful. On the other hand, if the value of relative risk is greater than 1, it means that more women are able to become pregnant under treatment. So the intervention is beneficial. The confidence interval is the uncertainty of the point estimate. And this, I would recommend that you calculate just simply using a statistical software instead of a calculation by a handheld calculator. And the meta-analysis has the ability to take all these data presented and combine them into a single result. You can see here in this meta-analysis that none of the single results of the trials are statistically significant. And this is because the confidence interval include the value 1. Now, in the first set of presentation, the studies were sorted by year of publication, but in the second example, they are sorted by study quality. And here you can see the figure already looks different. By year of publication, we cannot learn anything other than the effects are randomly distributed or virtually randomly distributed according to the year of publication. But by study quality, we can see that as quality changes from high to low, the result appears to have a particular trend. So you can see that the way you present data in a meta-analysis helps you understand what the combining of the results will achieve or can potentially achieve. So if the range of possible effect sizes is shown in this type of space described by the various circles, then if the truth effect size is expected to be in the middle, which we might call the effect size that is free of bias, then those outside the true, you could say, are the opposite of the truth, which is potentially untrue or lies, to put it in simple terms. Then combining the concept of reliability with validity, we can have a result that is valid and reliable. But potentially we can also have a result that is valid but not reliable, and the other options. And the validity is captured by assessment of risk of bias. So instead of year of publication, the risk of bias or validity assessment is a more suitable or informative way of putting data together. Here we show the same set of data again according to quality. And for meta-analysis, when we put these data together in a statistical software, the result produced is summarized normally in form of a diamond, where the points, if estimate, is in the center and the ends horizontally of the diamond represent the confidence interval. And here we see the confidence interval of the diamond. But look, it's important that we see it in light of quality. The high quality studies show that potentially the treatment is harmful and certainly is not statistically significant because the diamond is crossing the line 1. But it is the low quality studies which show a statistically significant result in favor of treatment. In this case, the conclusion cannot be that this treatment is effective. In this case, the conclusion got to be highly cautious because the high quality studies do not demonstrate any benefit. In the interest of time, simply to show you that small studies, i.e. studies that have wide confidence intervals, or the lines that cross the point estimate or the central point, are wider in small studies. Typically, in small studies, the expectation is that the studies with negative result or non-statistically significant result will be missing from the literature. So a study with a true result with a narrow confidence interval will be more likely to be published, but a study with a wide confidence interval with a positive result will be more likely to be published. This is called publication bias and is captured through an analysis called funnel plot analysis, shown here. So here you can see that possibly studies are missing from this part, and I can show it to you possibly through another example. When the studies are missing, this is called an asymmetric funnel plot. And this is because small studies tend to have a negative result and are not attractive for submission or acceptance, and therefore are not accessible when we carry out literature searches. This problem has got to be solved by what I covered yesterday, which is all studies should be prospectively registered. In this case, those undertaking systematic reviews will not end up missing studies, because if studies are registered, then systematic reviewers have the ability to search registers to find what studies have been planned. Here is a real published example of the same analysis that I showed you a moment ago, but an updated version of it. And here they have concluded that the treatment appears to be effective in improving pregnancy rates. However, if you look at this funnel plot, it is clear that there are some missing studies. I think you will obviously see that appear to improve, as stated in this publication, got to be a highly cautious conclusion. You cannot just jump from here to make a guideline in favor of the treatment. Here is another example where I want to emphasize the issue of quality. We are talking about randomized controlled trials, but if randomized controlled trials are not available, we tend to use non-randomized studies. In this case, I advise you to take more seriously the studies that are of a cohort design than of a case control design, because of the higher risk of bias in the case control design. Finally, once this statistical synthesis has been performed, a systematic approach is required to put the evidence together in order to reach a conclusion concerning a recommendation. And this goes through various steps, even in the case of randomized trials, which would normally be regarded as a high level of evidence. We got to consider other limitations. For example, is the outcome measuring the true effect indirectly? Or possibly there is inconsistency of results, or possibly there is heterogeneity that is important. In this case, the final quality assessment of the literature, even from randomized trials, may be low. And if the outcome assessed is not important, the strength of the recommendation may be weak. So from this, I take you on to preparing a systematic review for publication. In this case, I wish to highlight that a systematic review paper cannot really be a long thesis. It's got to be a short paper, potentially no longer than the size of a randomized trial. And therefore, the writing has got to focus on how a scientific article is constructed with the same structure as that for primary studies. I like to emphasize that the most important part is the abstract, because as we all know, most readers will not read beyond the abstract of a published article, including a published systematic review article. The title need to grab the attention, and in the case of systematic review, the term systematic review should appear in the title. Just like in the case of a randomized trial, the design randomized trial must appear in the title. The objective statement in the abstract should clarify the various elements of the research question, and the introduction should emphasize why the systematic review was required, in particular if no previous reviews exist or new trials have been published. Previous reviews may be assessed using the M-STAR quality checklist. The discussion is normally a hard part to write, but here you can compare the findings of your reviews with the findings of previous individual trials or previously published systematic reviews. Now, yesterday we talked about integrity of clinical trials. I emphasize that some trials are retracted because some misconduct or other flaws are found in them. But a lot of trials, even despite the flaws, are not retracted because they are not picked up in systematic review, in the process of peer review, or at present in systematic review, no methodology exists to capture them and remove them from the process of systematic reviewing. So here I present to you something entirely new. So I hope those of you who after this comstech workshop will proceed to undertake any systematic reviews of the trials you are undertaking will also focus on the aspect of integrity within systematic reviews. Your question should explicitly state that you will be mindful of integrity of the studies included. Retracted studies will obviously be excluded. Those with expression of concern would need to be assessed. Your search and selection strategy, which is step 2 of a systematic review, could specifically look for whether expression of concern exists about trials. The data extraction stage could involve collecting data that you can use for integrity assessment of the trials included. And your data synthesis can undertake pre-planned integrity-based subgroup analysis on the same lines that I presented concerning analysis of study quality. And finally, when you undertake grading, evidence should be downgraded if there are integrity concerns. And beware that following publication, if included trials are then subsequently shown to have some integrity flaws, then you should be prepared to issue updates of systematic reviews in light of new integrity concerns identified post-publication. I hope from what I have presented, you will be mindful that your own trials will meet the integrity requirements that were presented yesterday, and for which on page 88, from page 88, 89 onwards in your handout, you have an international consensus statement concerning integrity of clinical trials. So to conclude, the individual randomized trials may be of variable quality. Their results may be imprecise, or they may have wide confidence intervals. They may be published across the literature, perhaps in different languages, not necessarily captured through searches within just English language. And systematic reviews allow us to put these data together using meta-analysis if appropriate, and they allow us to design good trials for ourselves, and they also allow us to improve practice based on research. So with this, I conclude my presentation, and I'm happy to take questions both on systematic review as well as randomized control trial integrity. Thank you.
Thank you so much, sir, for that wonderful presentation. Because it's an online presenter, so the comment we have questions. Okay? So the other question that we have is around 3:30 will be for our guest here. So, sir, are you willing to take our question now?
I'm very happy to take questions. I noted that a colleague raised their hand in Zoom. So perhaps the colleague who raised their hand can ask their question if they wish. Please go ahead.
Participants, let's start with the online participant.
Dr. Sharon and Dr. Odoma have given some encouraging comments, so I'm grateful for your feedback. Okay, so Dr. Ratib is asking, what are the contents of a proposal for a systematic review? So I presume you are asking about how to create a protocol or a registration document concerning a systematic review. In this case, I strongly advise you that just as we use a clinical trial registry for prospectively registering our trial, we should use Prospero, a register for systematic review, and also we can use other platforms for registration. For example, Open Science Framework, an example of which I showed you yesterday concerning registration of the International Integrity Consensus. That platform can also be used for pre-registration of your systematic review proposals. Depending on the size of the systematic review, I believe we should also consider the possibility of publishing the systematic review protocol once it has been registered. And Dr. Maham is highlighting if there is any questions from yesterday, we can cover them, and has already put a question to me. I'll read that question out from the chat. The question says, what motivates researchers to falsify results, and what sanctions are available to those who falsify it. So, well, the simple answer is researchers are in competition either with each other or are under pressure in order to convince their employers or other peers that they are progressing fast, rapidly, or better than the competition. And if this puts undue pressure on them, and there are no mechanisms for oversight monitoring or checking to take place as the trials go on, then I think there is a possibility that they might take this easy route out to create, falsify, or fabricate data instead of making the effort to undertake the trial. In the example trial I showed you, you can see that undertaking a trial is an effortful activity. It's not just straightforward or easy. So I think I cannot say more than that concerning motivation. I think there is a responsibility for institutions and organizations to reward those who do well and to create some disincentives for those who don't do well. And I would see the sanctions in those light. I think instead of sanctions for detected misconduct, the focus should be on avoidance of misconduct from the beginning. We should nip the evil in the bud. So the courses of the kind that you are attending now, in my opinion, are far more important than thinking about punishment. This is the approach I suggest we should consider taking. Then there is a question by Dr. Najmuddin seeking the best duration of a clinical trial. Well, the answer is that it depends entirely on the question and the outcome and the duration of follow-up required. For example, in a cancer trial, the expectation would be that the follow-up should be at least 5 years. So I don't think there is a single formula that can help define duration of performing a trial. However, I ask you to consider the speed with which COVID vaccination trials took place. You may know that the average size of a properly designed COVID vaccination trial was around 25 to 30,000 randomized people. And these have been randomized very rapidly across many countries and continents in order to produce results rapidly, so that within 3 years, we are where we are today. So please consider that the duration of trial is also linked to how we can collaborate in delivering studies quickly. And for this, I encourage colleagues in the comstech platform to bring the 50 plus comstech countries together to undertake trials in a way that they can be performed rapidly and produce results quickly. Then there is a question by Dr. Imran, who is talking about, is it mandatory to register meta-analysis? At this time, it is not mandatory. But as I mentioned yesterday, the idea behind attending the workshop that we are in since yesterday and will complete today is not just to do what is the minimum requirement. It is for comstech investigators to show by example to the rest of the world that we are leading in good methodology. So I would very strongly say that even though registration is not mandatory, we should regard it as good research practice principle. So we should just automatically always register or pre-register our systematic review projects. I think you also ask about, other than Prospero, what platform exists, and Open Science Framework is one such possible platform. If your systematic review is about, for example, educational intervention, then there are platforms other than Prospero where you can register them. Cochrane, of course, also offers a registration and systematic review completion platform. Dr. Hakim has asked about research with respect to alternative medicine. I would recommend strongly that just because medicine is alternative herbal or non-pharmaceutical, it does not mean that systematic review and randomized trial methodology does not or should not apply to them. I believe that the randomized trial and systematic review methodology applies equally to all different types of interventions. There is a question from Dr. Yunus about post-market surveillance. So look, I have focused on presenting data emerging from randomized trials. So to Dr. Yunus and other colleagues, I emphasize to you that phase 4 is typically not a randomized study, it's an observational study. I think, given perhaps that I'm already overrun my time, perhaps Dr. Maham and other colleagues will gladly at another day organize something concerning surveillance where phase 4 type designs can be specifically discussed and developed. Dr. Imran has asked about study quality assessment. And I think for randomized control trial, the risk of bias assessment tool recommended by the Cochrane is now considered the industry standard. However, in some circumstances, it may merit some modifications. So on a case-by-case basis, you may or may not wish to use it exactly as it is prepared. My presentation will be available via Dr. Maham to anybody. And I have missed out on one comment, which is concerning the title of my book. I would be grateful if Dr. Maham is also able to just distribute the web link concerning my book. The title of the book is Systematic Reviews to Support Evidence-Based Medicine.
Thank you very much, Dr. Khalid, and I'll do that.
One of the speakers you say also wishes to ask a question, Dr. Maham. I'm happy if you have time available. I certainly have time available.
Okay, sure. Thank you.
Assalamu alaikum, Professor Khalid. This is Mr. Qadri, and it would have been wonderful to have you here physically with us. I think you've done an excellent job and contributed tremendously to our understanding of systematic reviews and integrity research. Just wanted to highlight one question that perhaps because you are part of this consensus group which produced the document in Egypt, if you are suggesting that the OIC countries perhaps can get together in performing collaborative randomized controlled trials, and certainly they can get together and perhaps help each other in conducting systematic reviews and meta-analysis as well. But the one limiting factor for researchers in the developing world and the comstech region is access to literature, comprehensive access to literature at all times. Would it be possible to perhaps suggest at the comstech level and at your level some kind of a digital access for researchers who should be able to get unlimited access to published and unpublished literature? That's a comment.
I 100% agree with you. Just as A randomized control trial, in my opinion, should be a priority for comstech. Systematic reviews and meta-analysis of randomized trials should also be a priority for comstech. I believe steps are being taken in this direction, and Dr. Maham, if she has more information from the head office, can potentially make some comments. But certainly, I hope she will take this feedback back to senior colleagues within comstech to promote this agenda. Because within healthcare and biomedical research, it is well established that systematic reviews are at the top of the evidence hierarchy. And this is what we should be investing in. So I appreciate the comment and the suggestion made. Thank you.
All right, sir, thank you. There is another question in the.
On the chat, there is a question about the minimum size for a clinical trial. So, well, first thing I would say is that the comments I make are not from the regulatory perspective. I imagine some of the presenters and some of colleagues in the workshop perhaps are thinking of the regulatory framework. I believe with respect to regulation, the industry that intends to undertake trial can seek advice from the regulator on sample size before commencing their trial. So that would be my simple advice to colleagues with respect to regulation. But those outside the regulatory framework, working within academic setting, for example, through universities, well, look, there is no single answer. This will depend very much on the topic, the importance of the outcome selected, and the expected effect size that is considered valuable. So it will vary from topic to topic, but also within the same topic from outcome to outcome. So I'm afraid I can't give a simple single answer. I can say that in my own clinical field, where I have undertaken trials in women's health, I believe that trials of any size smaller than 350 to 500 people will probably produce quite wide confidence intervals. And in this type of situation, clearly planning a prospective meta-analysis, which is to already have a team established of existing trialists who work together to bring the data together in the future when trials are completed, should be something of a collaborative good practice within comstech countries, for which, as the comment has been made earlier, comstech can offer a central platform. I'm happy to take another question or other comments.
Okay. All right. Yeah. Okay. There's another last question here. Okay.
Thank you so much for that very enlightening presentation. I just had one comment. This is something that I had some back and forth discussion with some of the panels, and I want to pick some of your opinion as someone who has been management for quite a number of years. When you make a decision to undertake your meta-analysis, for instance, in the absence of adequate primary studies, should it depend on the sample size, the few primary studies that you have? This depends on the number of studies that are available. That has been like a hard question for me to understand.
Okay, Dr. Maham, I apologize. Due to the sound connection, I could not pick up the question specifically. Are you able to write any key points which I may pick up? That will help me.
Let's take another question.
Okay, thank you, Dr. Maham. I await for the comment to be written.
No, sir, can you hear me? Okay.
Yes, please.
Thank you very much. Thank you for the extended and elaborate presentation. My own question to you is on meta-analysis. I'm going to split it into two. The first one is what is the average number of articles required to be reviewed for meta-analysis?
Okay, so your question is there a minimum number of studies that should be combined for a meta-analysis? So I think the simple answer is any more than one study can be combined in a meta-analysis. So 2 studies is the minimum required. But please be aware that just because a systematic review does not have many studies does not necessarily mean that it is a weak systematic review. Such a systematic review may in fact be highly informative about how future studies should be designed and conducted. So the value of a systematic review does not just depend on the number of studies it is able to collect.
Thank you very much, sir. The second question is this. Who are those that are eligible or the calibers of people that should review a meta-analysis article?
So you are asking, let me see if I understand this right. You are asking if it is possible to evaluate the quality of a published systematic review? Have I picked that up correct?
Yes, sir. When you submit the paper for review, who are those that are eligible, the calibers of people that should be allowed to review for systematic meta-analysis?
Okay, I am really sorry, I apologize. Because of not being able to hear the sound well enough at my end, I couldn't pick up the question, but if it could be written in the chat, again, I will be able to address it. But at the moment, I have 2 questions on the chat, which I have so far not addressed, and I'll pick them up. One of them is by Dr. Waqas Latif. And the question is, what is the difference between randomized clinical trial and randomized controlled trial? So I give you my personal perspective. A randomized controlled trial does not have to happen in biomedicine. It could happen in any other subject, wherever this design is relevant. For example, I understand in agriculture research, it is not uncommon that randomization of plots of lands can be applied to see with different fertilizer, for example, whether agriculture production is higher with respect to the intervention used. So the randomized control trial simply refers to the design where allocation is randomized, whereas the word clinical makes it more specific to healthcare. With respect to the next question, I think I already addressed the issue of minimum number of primary studies. Well, then there is another related question. Should the number of studies depend on the sample size of the primary studies? The answer is no. The idea behind systematic review is to collect all the possible studies conducted on a particular topic. And it should not depend on the sample size of the original primary studies. Of course, how the original primary study will contribute to the meta-analytic result will depend on its sample size. Then there is a question, who are the ones qualified to review meta-analysis? I presume you're talking about peer review of a meta-analysis paper. I think the answer then is that people who have the experience of undertaking meta-analysis can be peer reviewers. But look, just as we have a course 2 days on randomized trials, sure, one day comstech can organize a course and provide the resources required for undertaking reviews within its platform to facilitate the development and promotion of this type of research within comstech. Dr. Maham, Is there other other question? I have received another comment concerning the link of the book, so I take the liberty to, if you don't mind, add the link on the chat. It will take me a few seconds to just find it on the internet. Just give me a minute to figure this out. And here I'm able to provide you the systematic review book link within the within the chat of Zoom.
All right, thank you very much, Dr. Khalid, for your time and for your very informative talk. So with this, we just need to announce the service. Thank you very much, everyone.
Okay, thank you very much for the invitation.
Thank you so much, sir. So we have all benefited from the presentation, and you realize that the presentation generated a lot of questions. And what's the meaning of that? That means it's well presented and well understood. That is why we want to dig down to know more about the presentation. So we, at this point, can consent for giving us the opportunity to have session with these distinguished presenters. So thank you so much for that opportunity. As they go for lunch and prayer break, so exactly we expect to for them back here at exactly 2 p.m. So let's convey back here at 2 p.m. Thank you.
The lunch that came a bit late, I want to suggest we add more 10 minutes as we wind up. So we'll start exactly at 2:15, inshallah.
Okay. Bismillahirrahmanirrahim. First, I want to apologize because I know a lot of you are actually in the middle of your lunch. Still, I'll say that now I have a task of trying to deliver this talk while you're already digesting certain things, and then you have to digest this particular talk as well. Okay. So I know, but that's something I require as I have to ask a question. How many of you have actually been involved in any inspection or any you can say monitoring or quality inspection, things like that? Okay, so my talk will be more focused on this particular area because, as I have said before, that clinical trials, doing a clinical study is a very tedious process and it requires multiple steps to be very meticulously regulated. That regulation cannot be achieved only by the study team, which is actually executing. In fact, there might be lots of things or mistakes or errors or compliance issues which may be overlooked by the study team who is executing that particular study. So what is clinical trial monitoring? So Dr. Abdul Rashid is going to tell you much about ICH GCP guidelines in the next talk, but I'm taking the liberty of quoting certain sentences which applies to this particular talk, like the act of overseeing the progress of a clinical trial and of ensuring that it is conducted, recorded, and reported in accordance with the protocol, SOPs, and GCP, and any regulatory requirements. So this particular clause has been actually taken from the guidelines. That's why I stated directly here. So you can see that is this words like conducted, recorded, and reported. So the regulations of GCP applies to all these particular areas. So what's the purpose? Since we have been talking around for 2 days now about the protection of rights, and well-being of human participants, that's the main purpose of monitoring. Ensure consent in place for record access, then the trial data are accurate, complete, and verifiable. So, and the conduct of the trial is in compliance with the currently approved protocol, GCP, and all applicable regulatory requirements. So this is also being stated in the GCP guidelines. Now, what is types of monitoring? So with reference to a clinical trial, since trial is a long process, there'll be agencies which monitors your trial. First, you have to have a concept of some stakeholders which are actually involved in that trial. So there is just imagine a map, but there is a sponsor who's actually have a product who wants to study. Then there is a particular site or a hospital where that particular drug can be tested. And then there is another concept which I want to introduce, which we do not have talked much about, and that's the concept of a CRO. So CRO stands for Contract Research Organization. So it's a contractual research organization, and it's basically, it mediates or it helps in conducting the trial, but it did not become part of the trial conductance, but it mediates the trial process by engaging on one hand with the sponsor and on the other hand with the site, which is in our case, it's a hospital. So what it do is that particular CRO talks with the sponsor. If the sponsor says that this is a particular drug which I want to test in 35 hospitals, and say, for example, in a particular country, we do not have all the patients which can be enrolled from those 35 centers. So that particular CRO will now look for other centers in other countries. And in practical terms, what happens is, not all the time, and in not all the clinical research questions, all the study participants or the eligible number of participants are available in a particular trial site. So as Dr. Abdul Rashid has talked about in yesterday's his talk, that we have been engaged in multiple trials which were being conducted in multiple countries. The reason for that was that a trial is being conducted in so many countries so that a large number of participants can be recruited. Now, who identifies that this particular trial can be done in this particular country and in this particular country, these are the available sites or the hospitals which have enough capacity with reference to human resource, with reference to infrastructure, with reference to their facility to conduct a trial. So that is identified by the CROs. So the CROs now also, the sponsors delegates them the responsibility of not only just helps in conducting a trial, they also want that they should help them in actually identifying all the issues which can be there when a trial starts. So on that basis, the CRO routinely monitors how the trial is taking place. So there'll be a coordinating center of day-to-day monitoring that usually in a particular setup is at the site. Then there are data collected are consistent. This is what we usually do in a site, that the senior hierarchy of a particular site is routinely monitoring if the data is routinely collected is right. CRFs are being completed by authorized staff. No key data is missing. Data appears to be valid, and review of recruitment rate, withdrawals, and losses to follow up. Then there is a central monitoring, where we determine key eligibility criteria through collection of consent forms, scans, pathology reports, statistics, unusual patterns of data. Okay. The third type of monitoring, the on-site monitoring. This is particularly the type of monitoring which pertains to the CROs, where there is a staff training, access to necessary documents, confirmation of pharmacy and lab resource supplies, adherence to protocols in GCP, and and source data verification. So I'm going to talk more about source data verification in my later slides. So what a monitor is usually looking for when it comes to a particular site, they have an objective. They look for how the objective is, what's the study objective is, what's the purpose. Monitoring should be proportionate to the objective, purpose, design, blinding, size and complexity, outcome measure, risk. All these standards are laid down by the GCP. So normally what happens is when a particular patient is enrolled and his data is entered in a case report form, which is here as CRF, the CRF is completed and the data is entered, then that data goes into the software where there is a data cleaning and validation process. EDC stands for electronic data capture. So we have to train investigators. That training is usually conducted by a CRO. How the data has to be entered, when the data has to be entered, till what time the data has to be necessarily entered, because there is a timeline involved in it. Say, for example, I've experienced many times that if I have an eligible participant and that participant has been randomized to be enrolled in a trial, that randomization, after randomization, the relevant data has to be recorded within hours. At times, that window is around 6 hours. At times, that window is around 12 hours, but that window is not more than 24 hours. If by any case that window passes, then there'll be serious deviations from the protocol. And with reference to that CRF completion, the monitor visits and see whether that CRF has been correctly entered, fully entered. So, and there'll be, and plus, whatever the data has been entered, the source where data has been generated is the right data has been entered, because at times, whatever the data has been entered, if it's different from the actual site or the actual patient file, that will also create trouble. Okay, so do you have an idea of a CRF, case report form? Any idea? Okay, how many people have actually conducted a survey? Any surveys you have conducted or any feedback forms you have collected? Okay, so surveys basically involve a questionnaire. Feedback forms also involve a questionnaire. Certain questions are being asked. So CRF is technically a questionnaire. It involves certain questions relevant to the study. That data has to be entered. Just in terms that a survey has rather simple questions, and a trial CRF has a rather different type of questions. But those questions are tightly linked to, very meticulously linked to the trial objectives. Then the monitor also looks for if there is a problem in data entry, or the data has been entered in duplications. Then there'll be, and then at times they might need to scan the actual files of the patient. Then after data cleaning and validation, there are post-entry checks as well, which usually go as site we don't recognize, but the monitors and the sponsors are looking for those things. So what is post-entry check? So once the data manager cleans and validates database, problems such as missing values or inconsistencies are there, And at times the CROs then schedules a visit and see whether that data can be validated. If the queries had been addressed, all those queries are then addressed to the PI via paper query form, where, say, for example, if I have found some inconsistency in a hemoglobin entry, that's at times this happens is in actual file, the hemoglobin entry is like 10.9, while actually it was 11.3. So the monitor may ask why there is a difference of entry in it. And then the PI answers that in a reasonable way. What's the rationale? What was the, and that where then PI works on it and resolves the queries. Then that query resolution is put on a query form, and that monitor checks whether the query has been reasonably resolved. And that query form with the resolution sent back to data manager, and corrections are entered in database. So this is technically a cycle which every clinical monitor, other than their monitoring visits, also monitors remotely, and it's saying back and forth to the site for all these checks. So in a simple way, there is a review. After that review, there's planning. And after the planning, we implement those plannings. Then we check. And after checking, we review it again. And the cycles go on, and that's the goal of monitoring and audits. So the CRA, or the clinical research associate, which is usually hired by the CRO, will review study documents and may observe a study process to confirm adherence to GCP with reference to the data integrity and consent process and documentation and all other applicable regulations, particularly. So there are 3 types of visits. There is an inter-monitoring visit, for cause visit. For cause visit is like, as I've said in my previous table, that there might be a query, and for that, addressing that particular query, the CRO can schedule a for cause visit. Then there is a closeout visit. And then there is a site initiation visit as well. So in interim monitoring visits, which are routine visits by a clinical monitor for routine monitoring of a particular trial, they look for consent documents. And on the other hand, they also look for chart reviews, which are patient records, medical records. Plus, they also look into electronic file and paper file binder of those records. Plus, they also look for any reported serious adverse events, unanticipated problems which are happening in a trial, follow-up information, protocol deviations. So if a protocol says certain things and the PI has not performed on this, then that is considered as a protocol deviation. Then a research specimen review and investigational product accountability. So for consent from review, what a clinical monitor is looking for is whether this is obtained prior to initiation of study procedures. So they look for in source, like regarding when the informed process took place, they actually match when the randomization took place and when the informed consent process was done. So say, for example, logically, the randomization has to be after the consent took place. The clinical monitor usually looks for these things. So they look for date and time. Then they look for, in my previous talk, I was talking about clinical trial protocol amendments and new versions. So the new versions of informed consent can also emerge in a particular trial. So version numbers of informed consent is also important. Then study personnel conducting the discussion, who was actually taking the informed consent. Some person who was not related to the study, not identified by the CRO, and that person took the consent, and that is also something which the monitor look for. Then there should be an adequate time for review and consent questions, valid signatures and dates. So there's lots of trouble identifying. Lots of queries are there with reference to valid signatures and dates. And it has to be, the correct version has to be signed, including updates. So a clinical monitor with reference to source documentation will look for whether that particular source documentation, which in our case is the actual patient medical record, which can be either in an electronic form or in a hard copy, typical patient file. It has to be accurate, complete, and it must have all the information which clarifies why the patient was eligible in that particular study. And there should be, if any procedure has been done, which were required by the protocol, it has to be reported there as well. Proper documentation, reporting of side effects, serious adverse events in the source and data collection form database, accuracy of data recorded, and items directly entered in the database. So all these things are involved in with reference to source documentation. Then there is an investigator site file review. So investigator site file is something I also want to introduce with a term like TMF, trial master file. Then Dr. Abdul Rashid has repeatedly mentioned about investigative brochure. So these are the files which contains all the trial documents, which includes the protocol, the informed consent form, which is usually, particularly it's in English language, but it can also be in the local language. Then there'll be diary cards, which may be there for with reference to research question. And then there will be lots of other documents with reference to the conduct of the trial. So the monitor looks for all the essential documents are there. No expired document is there. And there's another sheet called site delegation log. The site delegation log actually ensures that the right people are in the study. It has to be ensured prior to the conduct of the study. Say, for example, a team is conducting a trial. So there's a principal investigator, then there is a co-principal investigator, then there is a trial pharmacist, then there is a trial nurse, then there is a research associate working on it. So all these persons names has to be there in the trial delegation log with their responsibilities mentioned. So and for unanticipated problems, the monitor will follow for previously reported side effects, serious adverse events, and any new events reported. Plus, one of the most important thing is IP accountability. So IP accountability is something monitor is looking for is whether it was storaged well, it was shipped well in the right way, the cold chain was maintained, and then if the trial has been completed, the IPs has to be returned as per protocol. So it also can perform during a monitoring visit all the IP counts for stock and return supply against all the dispensing logs and confirm proper disposition. With reference to protocol deviations, this is one of the major areas the monitor is looking for. It's identify potential deviations for discussion with the study team, whether deviations were from the document or they were not following the IRB guidelines, or there were deviations with reference to site personnel or established plans, and how they can be mitigated. Then there are for-cause visits, as I said. Lots of for-cause visits are actually for training or for any query resolution. Then there are closeout visits. When the trial is being finished, there is a particular closeout visit, which includes final consent form review, verification of essential documents, completion of data review and source verification, all the confirmation of serious adverse events being reported, all the confirmation of lab samples. So technically, closeout visits normally took a whole day, or maybe 2 or 3 days if the trial has a big number of enrollment. So during the monitoring visit, the SC will review the visit findings, develop action plan, and answer questions, and work with the study team to schedule a teleconference or whatever the medium is required to mitigate that particular action. Okay, so this particular site pertains to another term called auditing. So what's the difference between monitoring versus auditing? Monitoring is the act of overseeing the progress of clinical trial, while auditing is the systematic and independent examination of the trial-related activities in the documents. To monitoring is 100% source document verification of all the participants, while in auditing, snapshot in time of a subset of participants. So the audits can be done by the site itself just to ensure the quality. Okay. Then monitoring is to ensure that the study is conducted, recorded, and reported in accordance with, as I said before, for GCP. While auditing is to determine whether trial-related activities were conducted and recorded accurately, analyzed, and reported according to the GCP. Each protocol in monitoring will outline a data safety and monitoring process and plan, while we do not have any such plan with reference to audits. Some studies may require data safety monitoring board as well during the monitoring. So then there is another type of monitoring called site initiation visit. Technically, I personally don't consider it a monitoring visit because it's not a monitoring visit. However, it is still details about lots of things which overlaps with the particular responsibilities of clinical trial monitor, like when it is going to take place, if all the essential documents are in place, and IRB approval has been taken, and sponsor and CRO has relevant contracts placed well, and all that is documented, very well documented. So all these things are taking place with extensive documentation. Okay, so what happens in a initiation visit is the clinical monitor will review for study documentation. None of this will be filled, but still the monitor will be looking for that everything is in place so that the site at later stage cannot say, okay, we don't receive that or we don't know about that. So all the drug accountability form, PRFs, logs, and they will ensure that the site delegation log has been filled in that particular site initiation visit for all the signature log. And then also they give a monitoring plan of how frequently they'll be visiting that particular site. After that, they professionally provide a detailed letter telling what they have done and what they look for. So the time and scheduling of routine monitoring visits is a very variable thing because it depends on lots of variables, particularly the site performance, the number of queries which might be there, plus staff experience. So all these things and the complexity of the protocol. So it's something variable. But usually monitoring usually happens after every 2 or 3 months, but it can be even less. Generally, it's attended by CRO. As I mentioned, there'll be a CRA monitor, plus the personnel from site, which will be the PI, research nurse, protocol coordinator, data manager, pharmacist, and trial nurse. Okay, so one of the key responsibilities which monitoring, clinical trial monitoring involves is the documentation with the regulatory review. So they ensure that the regulatory binder is complete, like all protocol versions and approvals are there. The investigative brochure and versions are completely there. Lab certifications and normal entries are there. CVs and licenses of all the investigators with their financial disclosure is there. All the relevant IRB correspondence is there. Like if an amendment has taken place, well, is that documentation, is that notification to the IRB has been provided, and what the IRB has responded, all these things has to be provided in a documented form. So a clinical trial monitor all looks for these documents. Plus, most important, the serious adverse events and how that serious adverse events has been documented, how that has been reported, when it has been reported, then update of delegation log of responsibility, because at times there is an employee turnover. So at the start of the study, there were certain people, and during the course of the study, some people may left and some new people joined. So whether those people were properly included in that particular delegation log. Then in the regulatory review, they also look for amendments and serious adverse events and continuing reviews. They assure all participants and original consents are in the medical report. Okay. For source document review, a monitor looks for many things, lots of things. Particularly laboratory reports, because most of the errors arise in the lab entry. They also look for X-ray or scan reports. I have frequently encountered errors while during the course of my experience in reporting of scans. One radiologist says certain thing, another radiologist says certain other thing, and both of these findings might be recorded in the source document, but only one finding has to be there in the actual CRF. So there may arise confusion. Then there is a matter of then the physician notes are complete or not, because most of the physicians are lazy completing their documentation. But with reference to a particular trial, it's very much important that the physician notes has to be completed and it has to be comprehensive. Then if procedure has been done, then whether that procedure has been properly documented. Then there is a informed consent process. So that particular informed consent process has to be documented in the patient file as well. Okay, then review of pharmacy records. Okay, since we are usually dealing with drug trials, pharmacy records has a big role to play, and monitor looks for that. Whether the drug was properly dispensed, and therefore there is a drug accountability record form, whether the drug count is accurate and the disposal has been returned. In Pakistan, what happens is, I don't know about the rules here, but I could just give you an example of our country. The Drug Regulatory Authority of Pakistan, when they approve a trial, they also mandates that every drug who is authorized or licensed for a trial to come to the country, that number has to be provided to the regulatory agency. Say, for example, in a particular trial, 70 patients has to be enrolled. Therefore, the amount of drug which is required by that trial has to be known by the regulatory authority. And in that particular amount, the regulatory agency allows for that particular approval, they provide approval for that number. So in case, say for example, somehow few of the enrolled participants weren't able to receive that particular drug, and the drug was there as remained as a leftover, that data has to be mandatorily provided to the regulatory agency, and the regulatory agency then has a very meticulous protocol of disposing it. So the clinical trial monitor is actually looking for whether that remaining amount is there. So there are certain steps to make the monitoring visit go smoothly. This is usually required all our. This is something which we are memorizing all the time when our clinical trial monitor is coming towards our site, to arrange all the charts and CRF, and then we actually greet the monitor like a drug inspector, and we greet him. And we also look for, we have first hand of all our records. We memorize it and we try to provide all the details the monitor is asking for. Common mistakes, missing values, then there are range checks, like there might be a possibility that the CRO might have some other lab ranges, while your site might have another lab ranges, so that has to be coordinated. Then there is a logical date sequence, like say, for example, while running Chinese vaccine trials, we have recognized that they have a proper way of mentioning dates, which usually the Western trials do not have. So for running Chinese trials, we have to conform with that particular sequence of mentioning dates. We cannot use the Western format. And the clinical monitor looks for that. Plus, whether there are any missing visits or pages, the visits in compliance with the protocol, procedures performed in proper order, all these things which are common queries. Thank you.
All right, thank you so much. Please, let's reserve our questions to the end of the session. So we'll ask him all our questions. Thank you for that detailed lectures and presentation. So right now we call Dr. Abdul Rashid. He's going to talk on ICH guideline and GCP. So after that, then we have our discussion.
Thank you very much. Good clinical practices. Good clinical practices. Final GCP. Final GCP. Yes, this one. Thank you very much. Next, please. I will discuss the guidelines on good clinical practices. Yes. You know all about the good clinical practices. This is an international ethical and scientific quality standards. Number one, for designing, for conduct, recording and reporting of clinical trials where there is volunteer subjects are involved. The objective of ICH guidelines is to provide a unified standard for the European Union, Japan, the United States of America to facilitate the mutual acceptance of clinical data by regulatory authorities in their jurisdictions. So the whole globally, the scientists working in clinical trials, mostly they follow the ICH good clinical practice guidelines. There are some terminologies. I don't want to go into detail because we have shortage of times. I will go quickly because after some time we have a question and answer sessions. So at ADR, you know better it is in the approval of in the pre-approval clinical trial, clinical experience with a new medicinal product or institutional product in the new dosage, new usages, particularly in the therapeutic dose. The ADR is all nauseous or unattended response to a medical product, to the subject. Another is adverse event. This is adverse event is unwanted medical occurrence in a patient or clinical investigation subject or volunteers administered a pharmaceutical or investigational product. These are the different terminologies which are given in the good clinical practices. I don't want to go to the details. Now I come to the Independence Ethics Committee. Independent Ethics Committee, according to Pakistani law, this is an institutional, this is an independent committee or we can call it NBC net, and that is National Ethics Committee. This committee should be at the institutional level, at the regional level, at the national level, or even at international level. So this reviews the protocols and all the ethics of the independent clinical trials. These are the different terminologies which should be which independent ethics committee see the source of data, the source documents, sponsor investigator, standard operating procedures, sub-investigator, subject trial, subject title, and different aspects. Institutional Review Board, they have in good clinical practices, ICET guidelines, I give the responsibility. What is the responsibility of Institutional Review Board? What should be the compositions? What are the functions and operations? And how the IRB will work, and its documentation and record. So all the standards, international standards, they have been mentioned in ICH guidelines. Mostly the regulatory authorities are the sponsors. They follow these standards. Sponsors, I don't want to go into detail. There are the different responsibility of the sponsors, which is given ahead of you. That is, they initiate sponsors. Basically, it may be the individual, it may be the institution, it may be the organization, or any ministry which initiates, which have the management, and most important is sponsor the finances and give the finances. Sponsor is responsible for initiation of clinical trials, for the management of clinical trials, and for financial support. These are the different. There are different responsibilities of the sponsor. I do not go into detail because in the morning I have already mentioned in detail. Clinical trial protocols. This is very important. The clinical protocol must mention in clinical protocol, there must be the objectives of the clinical trial, the design of the clinical trial, methodologies, and other relevant considerations. It must contain general information, a trial design, selection, and withdrawal of the subjects, and different, also this statistically different parameters. Clinical trial protocols, these all are. I want to discuss about the investigation brochure. It contains all the details, all the standards of investigation drug, the dose, the dosage regimen, and the duration of the treatment by the investigation guard. All is given in the investigation brochure. This is the responsibility of the sponsor, that he should give full details about the investigation brochure. These all are in investigation brochure. There must be the general consideration, the physical, chemical, and pharmacokinetics parameters, also the non-clinical study data, also the toxicology, effects in humans, pharmacodynamics, safety and efficacy, marketing experience, and summary of the data. It should all be mentioned in the investigation brochure. Thank you very much. Now please go to the safety and vigilance of investigation. As the basic understanding, I have mentioned many things in the morning, so I don't want to go to detail in the good clinical practices. I mentioned briefly. Safety. Last. Safety and vigilance of investigation product. As I already mentioned that during COVID, I remain chairman of clinical study committee and I've started clinical trials in Pakistan. The same I was also the head of Pakistan National Pharmacology Center, and we have made mandatory and we collected advanced event following immunization of the essential drugs, about 60,000. And that was our success story, that in one year and a half months, we collected more than 60,000 advanced event following immunization of vaccines. And in 2022, I published 2 research publications on the safety of the COVID vaccines. Investigational product is an active substance or the placebo being tested during a clinical trials in a specific dosage form. These are the ethical considerations for clinical trials regarding safety management. There must be some the background and introduction of the any investigation product which is available in the previous data and the previous record and the previous research papers. They must be recorded. The stakeholders are the patients, regulatory authorities, and public health community. Another stakeholder is the investigators, institutional ethics committee, and institutional review board. They are also the investigators. Data and safety monitoring boards, they are also the investigators. Pharmaceutical companies and their representatives, they are all stakeholders during the safety management of clinical trials. And evolving regulatory and societal demands, there must be considered primary and secondary, primary and confidentiality of personal data. Informed consent, I've already mentioned in the morning that informed consent must be in the local language in the same country. Transparency in availability of clinical trial results. There must be the accurate data, accurate record. There should be no fake data, and otherwise it will affect on the safety of the investigation product. And the other issues that may be many. Good pharmacovigilance practice. How many personnel know about the good pharmacovigilance practice? Can you raise your hand? Good pharmacovigilance practice. Anyone? Anyone who knows about adverse event following the immunization? None. So, good pharmacovigilance practice. This is nowadays priority in the whole world, especially during the COVID, especially during conducting the clinical trial. There are some systematic approach that when you start the clinical trial, you start the monitoring of safety or adverse event of the subjects. Also establish a procedure for how to conduct the adverse event of how to conduct the serious adverse event. Maybe there is the mortality and morbidity of the investigation trial. Must establish a multidisciplinary safety management team. You can also call this multidisciplinary safety management board, or you can say multidisciplinary safety management committee. You can say any word. Establish a project management functions, determine background data, ensure the accessibility of the data. The sponsor, the regulatory inspector, or IRB, they must have the access to the data. This is very, very important. And it about the safety of the investigation product. Develop a proactive approach. The clinical trial must be that there should be no chance of damage to the subject. There should be no harm to the subject. There should be no serious adverse event to the occur to the subject. Establish a time frames and milestones Also, there should be decision making when there is any serious adverse comes. You must have the advisory board and the experts from different field, that is toxicology, epidemiology, public health, pharmacists, clinical pharmacists, clinicians. They all should be available in the advisory board. These are the different good clinical practice. I don't. One thing is very important: how you collect collection and data of safety and data monitoring board. This is very important. And who will collect? The most important to collect ADR or serious adverse event is can be collected from the hospitals where patient is there. Who is there? Investigator, clinician, nurses, and the investigator. They are the persons who can collect the data of adverse event. What it should be the what are the general principles? Data that should be collected and evaluated for safety will depend on the design of the clinical trial, but may include adverse events. It is the experience suffered by the patients, also the laboratory values, pharmacokinetic data. Pharmacokinetic data include absorption, distribution, metabolism, and excretion, results of mental and physical experimentations, spatial study data, patient demographics. These are the general principles on that the safety data can be collected. This is also the continuation of how you collect the data. Casualty assessment, if there is side effect, say in one city, say in Kampala, 20 same side effects has been reported. So there must be casualty assessment. What are the, you have to nominate some experts. They will analyze what is the root cause analysis of that event. Either it is due to the administration drug, either is the error of the doctor, either is it error of the nurse, or it is error of the pharmacist. So you have to do the casualty assessment. What is the actual cause that patient has been harmed? You must sign, this is the during diagnosis or sign and symptoms. You will have to see that one. Also, you must see the adverse event of special interest. You must consider that one. Also, you have to consider when there is a serious adverse effect, that is, what are the laboratory chemistry measurements. You must be, that it should be the use of clinical lab tests as surrogate makers for toxicity in early clinical studies. So they play a very important role. You must see that the side effects are occurring and morbidity and mortality is there are as a efficacy endpoints. In Pakistan, one of the hospitals, that is Aga Khan Hospital, there was 2 patients that were dead. So we have to see that this is due to the medicine, that is due to the investigation drug, or its efficacy is not so much. So we have to consider that it is due to the morbidity or mortality or efficacy endpoint. General concentrations, there are different ways you can collect ADR on paper, electronically. In Pakistan, we have the mechanism to collect adverse event formalization or adverse ADRs on the paper, through WhatsApp, through email, through fax. Even we have med safety mobile application. Our 220 million population, Pakistani population, they can report the ADR either for the adverse drug reactions or other routine introduced medicines on their through their mobiles. Either it is the iPhone or it is Android phone. We have the med safety application. 22 million population, they can report the ADRs. And another thing is, we were the 9th country which have started med safety mobile application. Up till now, total 15 countries, they have started med safety application to report ADR or safety of the medicine.
Serious and other important adverse events that you have to consider when you start, when you take the consent form from the subject, the ADR reporting should be. And when it is closed and it is safely used by the patients till the end of the closing, till the closing of the clinical trial, you must collect data of ADR or adverse event following initiation of an investigation trial. I have to go to the quickly clinical description of the adverse reactions. There is coding procedure and decoding procedures. Sometimes patient don't know, even doctor don't know, even investigator don't know, even principal investigator don't know, pharmacy don't know. Only one personnel, either he's an IT personnel or other clinicians, who is specified only for coding and decoding procedure, and that is also given in good clinical practice. Identification and evaluation of risk from clinical trial data, you have to assess that after collecting the ADR safety, you can assess this is a safe medicines or not. You also have to consider and apply statistic parameters, and then you have to decide that this medicine is safe or not. The regulatory reporting, every principal investigator is bound to submit the ADR or serial address even following immunization every month, and within the mid of the clinical trial and after the closing of the trial. So this is a regulatory requirement. Principal investigator have to submit that. With this, thank you very much. Any question, please? You know, adverse event following immunization is data is collected only for vaccine, biological, and sera. And adverse event, serious adverse event, and ADR are collected for medical devices, upper medicine, homeopathic medicine. That's an answer. Repeat, करें? Yes, of course, if the patient is damaged, even one part of his part is damaged due to initiation drug, the subject is insured, and it is the full responsibility of the sponsor to give the remuneration, and he is fully responsible to give the full treatment. This is given in the SOPs. During COVID, we have seen that even the patient who is at home, in Pakistan, at the time of vaccination, the vaccinator give him one phone number. He advised them that if at home, maybe today, tomorrow, or day after tomorrow, you feel any adverse event following immunization, that is AEFI, please call in this number. Our ambulance, our vehicle will go, and they will bring it to the hospital and give the full treatment. Moreover, definitely the subjects are insured in every clinical trial, and remuneration is given to them. Thank you.
Thank you for the. What do you could clarify on the rules where during an RCT, the effects are so spectacular? What are the rules on interim analysis and stoppage of the trial where you've detected the effect is so spectacular you don't need to continue?
Okay, so the question is, if I can repeat the question, that during a trial, when you're doing interim analysis, what are the rules for stopping a trial for either suspected serious adverse events or a number of side effects, or for efficacy or anything? What are the stopping rules? So the stoppage rules have to be predefined. when you're planning an interim analysis, and the Data Safety Monitoring Board actually then sitting with the investigators, like there's a predetermined thing in the protocol that at perhaps 30%, 50%, 75% of the trial, the interim analysis would be done. So usually it's like 25, 50, and 75% timing. And when the interim analysis is done, obviously things are assessed for futility, that are you, for adherence to the protocol, for the recruitment, looking at if the recruitment is appropriate or not, looking for futility, that you have not seen any benefit and you're already 50% into the trial, or you're looking for serious adverse events or serious unsuspected adverse reactions or adverse drug reactions, and or you are looking for benefit. Now, what happens is that a lot of times the clinical trials, when they move, they are moving in a zigzag fashion. So they go a little initially, when the numbers are small, they may go towards the right. When the numbers go a little bigger, towards the left, and like that. But then after a little while, the trial develops some stability. That stability may come after 30, 40% usually. So if people stop the trial too early, they may sometimes stop it too early for benefits, or sometimes stop it too early for harms. So the good idea is to let the data get stable after a certain number of patients recruited. And then there's a predefined endpoint as to what would constitute something. Trial stoppage rules are pre-specified, and that is already known to the Data Safety Monitoring Board, the sponsor, and the investigator. The only problem is that once you do interim analysis, then you have to do an adjusted analysis, and that adjusted analysis is a statistical thing where you look at 3 or 4 different methods. One is Habertle Peto, another one is O'Brien or Fleming, another one is Pocock, and all of them have a certain alpha spending error, they say, the false positive rate. So you have to deduct that out of 0.05, and it may come down to something like 0.046 or something would become the p-value. So for every single serious event, which is a serious morbidity, or majority of the subjects having significant benefit, then the rules are that they can only recommend. They don't stop it. They are an advisory board. So the eventual responsibility for trial conduct is with the sponsor. So the advisory body will tell the investigator will report to the IRB will, and the investigator will reach the sponsor, and then the sponsor has to look at that data and say, yes, one should stop or not. So this is a responsibility of good clinical practices at all levels, and I think that is part of that. And pharmacovigilance will pick up a lot of those things, because if the serious adverse reactions are being reported from one site, one particular site, then even the principal investigators, others, when they see a signal from one particular site in the world where maybe because of biological variability or difference in metabolism, things are getting out of hand, then that site will stop everything till they can clarify what were the events due to, because then you have to find out, was it an attributable risk or not? I mean, like, was it something that can actually directly be attributable to the drug or not? Because sometimes you may have adverse events which occur just during the course of a trial, but they may not be directly attributable. So somebody has to decide within pharmacovigilance, within this trials or Data Safety Monitoring Board, that yes, this is probable, this is possible, or this is highly unlikely to be related to the drug, and do that. So if somebody during the trial, I'm enrolled in a vaccine trial and I go around and fiddle with that electrical outlet and I drop dead, then, I mean, like, they will find out that this is what was being done. So I mean, that's obviously is an adverse event that occurred, but not directly attributable. So I hope that answers your question. So the floor is open for questions. I just want to make one comment about the GCP. I think it's important for all of us sitting out here to understand that the good clinical practice that we go through requires a certification. So anybody who's conducting a clinical trial, international or national level clinical trial or multi-centric clinical trial, they all need to be the key personnel, the principal investigators, the nursing related, the pharmacy related personnel, the clinical research associates, all of them involved in the trial. The key investigators and co-investigators need to have GCP certification, which can be obtained from a variety of open source sites. So the IC at GCP do not endorse any GCP program that says that this is from the ICH GCP, but their content is used by several agencies which are doing it, some charging a fee. A very useful site internationally is with the US NIH NIDA. So they have a National Institute of Drug Abuse network attached to the NIH website. So that NIH NIDA site has the GCP certification course. It's free. So you have about 11 modules in there, and every module has 5 questions. You answer them about 3 or 4 out of 5 and all that, and eventually get a certification probably the same day. So that certification remains valid for 3 years. So one needs to have the GCP certification. So because that ensures that all the things we are talking about today are actually put into practice, and you understand the importance of each aspect of that. Yes, please.
Yes, assalamu alaikum. Yes, thank you very much. My question is still on this issue of adverse event and serious adverse events. First, I want you to clarify, when do we say an event is serious? That is one. Then, using this case of vaccine, which most people have taken, some people are still developing some adverse effects. When do we say, do we wait till when somebody dies before we now trace it to the vaccine that has been taken? When do we now say that this event is due to the vaccine taken? Thank you.
Let me answer the first part and let Dr. Rashid answer the second part, that there are certain defined things which clinicians recognize. So the clinical team recognizes that whenever you're doing any surgery or any intervention in the hospital, the similar thing you're doing when you're taking a vaccine. And you always have certain things which are basically classified as sentinel events. Those sentinel events are things which can be, of course, at the extreme death. It could be a myocardial infarction. It could be a loss of limb. It could be a loss of eye. It could be a serious lab result abnormality. It could be a serious radiological abnormality. Anything which is of concern requiring hospitalization automatically will be a serious adverse event. And anything less than a hospitalization would be somewhere in that range. But it all depends on how a clinician views the severity of an event. But certain events are completely well defined. Maybe Abdul Rashid Saab and Dr. Ahmad can add to that.
I want to add one thing, that for serious adverse event, which is considered during the clinical trial, if it is harmed by the drug and serious adverse event, there are 4 things. Number 1, that there may be the chance of mortality. The second is there is chance of morbidity due to that medicine. The third one is prolongation in the hospitals. If it's already in the hospital, so due to adverse event, his stay in the hospital is prolonged. The same is with this, that if the subject is not hospitalized, but serious adverse event occur, he is in the home and he is shifted to the hospital and he required hospitalization. This is one of the third reason for serious adverse event. Another is any persistent abnormality, any persistent sign and symptoms. They occur, then this is considered as a serious adverse event. So we have a form, ADR form, which is approved by the WHO and Pakistan, and hopefully the Ministry of Health, Uganda is also following the same ADR for yellow form. So we have the form. If it is routine ADRs are due to the medicines, it's okay. We have signs, symptoms. We are the investigator. He fills all the form. But if it is a serious adverse event, I mentioned 4 or 5 things, they will be considered as a serious adverse event. Moreover, if the cases are more, there is the casualty assessment where they make a committee for investigation
what is the root cause analysis? What is the actual cause that this serious adverse event has been done by the investigational drug, or the mistake of the clinician, or the mistake of the nurse, or there is a relationship with the investigation drug, or there is the mistake of the pharmacist. So these events, they consider all the things and they decide that this is a serious side effect or the routine use. Thank you. Any more question, please? My request will be that along with clinical trials, I must request you people, you belong to the healthcare professionals, you are healthcare professionals, you must think about also the pharmacovigilance, because the medicines which give the benefit, which treat you, which prevent the diseases, but there is a serious issue throughout the world. WHO is giving importance. Pakistan is also giving importance. When you take the allopathic medicines, you must consider the adverse drug reaction. That is a pharmacovigilance throughout the world. Another thing is, which is very important, the patient safety. In developing countries like Pakistan and Uganda, patients are suffering a lot of due to antibiotics, due to surgical procedures, due to sepsis, due to infection prevention control, due to radiological errors, due to pathological errors, and embolism. So nowadays, main priority in the advanced world is given on patient safety. Moreover, there are a lot of non-communicable diseases. 65% are patients, they are suffering from cardiovascular diseases. They are suffering from stroke. They are going to death. Cardiovascular diseases, the patient, 65% are dying due to cardiovascular diseases. So these non-communicable diseases, they are very important, and these should be given consideration in Uganda. We also I will prefer that there should be another training on public health in primary health care, inshallah, in IU and at the hospitals. So if there is any question regarding 2 days, then 3 resource persons are there. Please, any question?
Yeah, there's one question here.
Thank you very much. Of course, we know that effects of medicine sometimes they immediate,